Today we are going to tell you about the dawn of human olfaction, when our ancestors first sensed the allure of good smells and the warning of bad ones, how they learned to interpret these invisible signals, mastered their power, and the timeline and places where this sensory journey unfolded. Welcome to the first part of our exploration into one of humanity's most ancient and enigmatic senses, the sense of smell. This opening chapter will journey back to the very roots of olfaction, tracing its origins not just in our species, but in the primordial soup of life itself. We'll uncover how this sense emerged as a survival tool, how early organisms began to distinguish between enticing and repelling odors, and set the stage for how humans would eventually harness it. This is a story that spans billions of years, from microscopic beginnings to the complex neural pathways that define our perceptions today. As we delve in, imagine the world not through your eyes or ears, but through your nose, a portal to memories, dangers, and delights that has shaped our evolution in profound ways. To understand when humans first sensed good and bad smells, we must start at the beginning of life on Earth. Olfaction, or the sense of smell, is phylogenetically the oldest sensory modality among all living organisms. It predates vision, hearing, touch, and taste by eons. Scientific evidence suggests that chemical sensing, the precursor to smell, emerged in single-celled organisms around 3.5 billion years ago during the Archean Eon in the warm, chemical-rich waters of ancient oceans. These primitive bacteria and archaea didn't smell in the way we do. They detected dissolved molecules in their watery environment to navigate toward nutrients and away from toxins. Good smells were the molecular signatures of food sources like sugars or amino acids, drawing them in for sustenance. Bad smells were signals of harmful substances, such as heavy metals or acidic compounds, prompting evasion. This basic chemosensation was a game changer for survival. In those early seas, where light barely penetrated and sound waves were muffled, chemicals were the language of life. Enzymes on cell surfaces acted as primitive receptors, binding to molecules and triggering responses like movement or reproduction. Fossil records from ancient stromatolites, layered rock formations built by microbial mats in Western Australia, hint at these communities thriving in environments teeming with volatile compounds. Here in what is now the Pilbara region, life learned its first olfactory lessons: attraction to the beneficial and repulsion from the detrimental. As life evolved into multicellular forms around 600 million years ago during the Ediacaran period, olfaction became more sophisticated. Early animals, like the worm-like creatures fossilized in the Burgess Shale of Canada, developed specialized cells for detecting chemicals. These were aquatic environments, so smell was more like taste, dissolved particles in water stimulating sensory pits or tentacles. Good smells signaled prey or mates, bad ones warned of predators or decaying matter. The Cambrian explosion, around 541 million years ago, accelerated this. Trilobites and early arthropods in what is now the House Range of Utah left traces, suggesting they used chemical cues to hunt and avoid danger. Paleontologists infer this from the positioning of sensory organs on fossils, which were optimized for detecting waterborne odors. The real leap came with the transition to land around 400 million years ago during the Devonian period. Fish-like amphibians, such as the Tiktaalik rosy discovered in the Canadian Arctic, began crawling onto muddy shores. Their olfactory systems adapted from sensing water-soluble chemicals to volatile airborne ones. This shift marked the birth of true smell as we know it. In these early tetrapods, nostrils evolved not just for breathing, but for capturing scents carried on the wind. Fossilized skulls from Greenland show enlarged olfactory bulbs, indicating a growing reliance on smell for foraging in unfamiliar terrestrial landscapes. By the Permian period, around 300 million years ago, reptiles dominated and olfaction refined further. In the Karoo Basin of South Africa, fossils of synapsids, mammal-like reptiles, reveal complex nasal passages. These creatures distinguished good smells like fresh vegetation or potential mates from bad ones like rotting carcasses or volcanic fumes. The sense was mastered through neural expansion. The olfactory epithelium, a patch of receptor cells, grew, allowing detection of thousands of odorants. Genetic studies of modern reptiles trace this back, showing odorant receptor genes multiplying during this era. Mammals emerged around 200 million years ago in the Triassic, in regions like the Isquigualasto Formation in Argentina. Early mammals, small and nocturnal, relied heavily on smell to evade dinosaurs. Fossils from China's Lufeng Basin show tiny skulls with prominent olfactory regions. Here, the distinction between good and bad smells became hardwired for survival. The sweet aroma of fruit signaled nutrition, while the acrid stench of predator urine meant flight. Learning occurred through trial and error, associating a smell with a positive outcome like a meal reinforced attraction, while a negative one like illness built aversion. Primates, our direct ancestors, appeared around 65 million years ago after the Cretaceous extinction, in the lush forests of what is now Madagascar and mainland Africa. Early primates like the Adapids, fossilized in Germany's Messel pit, had keen olfaction for navigating dense canopies. Good smells guided them to ripe fruits and flowers, bad ones to spoiled food or threats. As primates evolved color vision around 55 million years ago in African forests, some olfactory genes pseudogenized, became non-functional, but humans retained about 400 functional receptors, allowing us to discern millions of scents. Hominids diverged around 7 million years ago in Africa's Great Rift Valley. Sahelanthropus tichadensis, found in Chad, likely used smell to forage in savannas. By 4 million years ago, Australopithecus in Ethiopia's Awash Valley distinguished edible plants from toxic ones by odor. This learning was social. Groups shared experiences, teaching young to recognize scents. Mastery came with tool use around 2.6 million years ago in Olduvai Gorge, Tanzania. Homo habilis might have smelled smoke from early fires, associating it with safety and cooked food. Homo erectus 1.8 million years ago in Kenya's Kubai Fora migrated out of Africa using smell to track game across Eurasia. Fossils from Java show enlarged brains with olfactory processing areas. Good smells like fresh water or herds, bad like decay or ash from volcanoes. Around 300,000 years ago, Homo sapiens emerged in Morocco's Jebel Irhoud with a sense of smell fine-tuned by genetics. We learned through culture, elders passing knowledge of medicinal plants by their aromas. Archaeological evidence from Kafse Cave in Israel, 100,000 years ago, suggests early humans used scented resins in burials, mastering smell for ritual. By 40,000 years ago in Europe's Lascaux caves, cave art depicts animals whose scents were familiar. In Australia's Arnhem Land, rock art from 20,000 years ago implies olfactory hunting knowledge. This evolutionary arc from ancient oceans to African savannas laid the foundation. Good smells drew us to life-sustaining resources, bad ones shielded us from peril. Learning was instinctive at first, then experiential and social. Mastery evolved from survival to symbolism, setting the stage for cultural perfumery. In the vast silence of ancient seas, long before any creature crawled onto land, the sense of smell was already alive and working tirelessly. This second chapter plunges us into those underwater worlds where chemical detection first became refined, where the line between smell and taste blurred completely, and where the earliest complex animals began not merely to react to odors, but to actively seek them, avoid them, and even use them to communicate. Here in the oceans of the Cambrian, Ordovician, Silurian, and Devonian periods, the foundations of olfactory mastery were laid, foundations that would carry forward all the way to the noses of our own species. Picture the shallow, sunlit waters covering much of what is now northern China around 520 million years ago. The seafloor teems with strange life during the height of the Cambrian explosion. Anomalocaris, a meter-long predator with grasping appendages, sweeps through the water column. Its head bears a pair of large, stalked eyes for vision, but equally important are the small pits and grooves along its mouthparts and antennae-like structures. Paleontologists who have studied exceptionally preserved specimens from the Zhejiang biota in Yunnan province interpret these as chemosensory organs, early versions of what would become olfactory receptors. Anomalocaris did not simply see its prey, it smelled it. The faint chemical trails left by trilobites scuttling across the sediment, the amino acid signatures leaking from injured soft-bodied worms, the subtle gradients of dissolved organic matter, all these became detectable cues. A good smell here meant opportunity, a wounded animal leaking proteins, an aggregation of plankton-rich water. A bad smell meant danger, the acrid compounds released by certain idarians when disturbed, or the bitter alkaloids exuded by toxic sponges. These early arthropods learned through simple association. A larva that followed a pleasant chemical gradient toward a microbial mat found food and survived to reproduce. One that swam into a cloud of repellent molecules, perhaps hydrogen sulfide from anaerobic decay, retreated and lived another day. Over countless generations, this trial and error built instinctual preferences. No conscious thought was involved. The nervous system wired itself so that certain molecular shapes triggered attraction pathways while others activated avoidance circuits. The number of odorant receptor types began to increase. Genetic reconstructions suggest that the Herbaliterian, the last common ancestor of most bilaterally symmetrical animals, already possessed several dozen chemoreceptor genes. In the Cambrian oceans, that number started climbing rapidly in certain lineages. By the Ordovician period, 470 million years ago, the seas covering what would become the Appalachian Mountains in North America supported diverse ecosystems of jawless fish, nautiloids, and early eurypterids, sea scorpions. Eurypterids, some reaching 2 meters in length, left spectacular fossils in the Silurian rocks of New York State and Estonia. Their paddle-like appendages and clustered sensory pits on the head indicate sophisticated chemical detection. These creatures were ambush predators and scavengers. They could follow scent plumes over long distances in murky bottom waters where vision was useless. A good smell might be the iron-rich hemoglobin leaking from a dying orthocone nautiloid. A bad smell could be the defensive secretions of certain trilobites that contained quinones, sharp, irritating compounds that modern analogs still repel fish. Learning here was still largely hardwired, but some flexibility appeared. Juveniles that survived an encounter with a toxic prey item were less likely to pursue similar chemical profiles later in life, suggesting rudimentary aversive conditioning even at this remote stage of animal evolution. The Silurian and early Devonian, between 440 and 390 million years ago, brought another leap. In the lagoons and river deltas that left behind the famous Rhynie chert of Scotland, the first land plants appeared, simple Cooksonia and Asteroxylon, while the seas nearby teemed with placoderms and lobe-finned fish. Among these lobe fins, forms like Eusthenopteron from Miguasha in Quebec possessed nasal sacs connected to the oral cavity. Water flowed in through external nostrils, passed over olfactory lamellae, folded sheets of receptor tissue, then exited through internal openings. This dual system allowed detection of both waterborne and, crucially, airborne volatiles when the animal's head broke the surface. The distinction between smell and taste began to sharpen. Good smells now included the fresh, green volatiles released by emergent marsh plants. Bad smells included the sulfurous stench of anoxic mud or the alarm pheromones released by schooling fish under attack. These early tetrapodomorphs learned olfactory categories through repeated exposure in complex habitats. A fish that repeatedly found safety in weed-choked shallows rich with plant terpenes would return to similar odors. One that suffered gill damage from swimming into a stagnant pool heavy with hydrogen sulfide developed strong avoidance. Social learning may have begun here as well. Schools of early fish often synchronize behavior using chemical cues. When one individual releases an alarm substance after predator contact, the entire group scatters. Survivors associate that sharp, distinctive odor with danger for the rest of their lives. This is cultural transmission of olfactory knowledge at its most elemental, no language, no teaching, just shared physiology. Around 375 million years ago, in the subtropical floodplains that are now Ellesmere Island in the Canadian Arctic, Tiktaalik rosy lived. Its fossilized skull shows greatly enlarged olfactory regions compared with fully aquatic fish. The nostrils are positioned high on the snout, ideal for sniffing air when the animal rested with its head above water. Tiktaalik hunted in shallow, plant-choked channels. Good smells guided it to concentrations of small prey fish or crustaceans. The aroma of crushed vegetation signaled nutrient-rich feeding grounds. Bad smells, rotting carcasses, low oxygen water laden with methane and sulfide, prompted it to move on. Here, for the first time, we see evidence that an animal could compare olfactory information across time and space. Tiktaalik likely remembered productive hunting spots by their characteristic scent profiles and avoided previously dangerous zones. This spatial olfactory memory represents an important step toward mastery. As true tetrapods emerged in the late Devonian, creatures like Acanthostega and Ichthyostega from East Greenland, the transition to land accelerated olfactory evolution. Their nostrils became true external nares, no longer solely for water flow. The olfactory epithelium thickened, receptor cells multiplied, and the olfactory bulb in the brain enlarged dramatically. These animals still spent much time in water, but they could now detect airborne molecules. A good smell might be the sweet, honey-like fragrance of early seed ferns or the yeasty odor of fungal mats. A bad smell could be smoke from distant wildfires or the bitter tannins of unpalatable plants. Learning became more individual and experiential. An animal injured by a toxic millipede would forever avoid similar pungent defensive secretions. A female might follow the musk of a receptive male across hundreds of meters of floodplain. By the Carboniferous period, 350 to 300 million years ago, vast swamp forests covered equatorial regions that are now Europe, North America, and China. Amphibians diversified into large predatory forms like Eriops from Texas red beds and smaller, more terrestrial types. Their olfactory systems were now fully adapted for air. Nasal passages contained turbinates, thin bony scrolls that increased surface area for receptor cells. This allowed finer discrimination. Good smells included the resinous aroma of chordates trees, the fruity volatiles of early gymnosperm cones, the earthy scent of fertile soil after rain. Bad smells were the acrid pyroligneous acid from smoldering peat fires, the ammonia-heavy odor of urine-soaked ground near dens, the putrid gases of anaerobic decay beneath leaf litter. Learning in these environments was intensely social. Many carboniferous amphibians gathered in breeding ponds during wet seasons. Males released pheromones to attract mates. Females distinguished preferred males partly by olfactory signature. Juveniles learned which plant odors signaled safe shelter and which warned of hidden predators. Some species may have used scent marking to delineate territories, just as modern salamanders do. A male that survived repeated border skirmishes would associate the mingled scents of rival urine and blood with conflict and adjust his movements accordingly. This marks the beginning of olfactory communication as a tool for social mastery. In the Permian, 299 to 252 million years ago, the supercontinent Pangaea dominated. In the Karoo Basin of South Africa, therapsids, mammal-like reptiles, proliferated. Fossils of cynodonts show extraordinarily complex nasal cavities with elaborate turbinates. These structures warmed, humidified, and filtered incoming air while vastly expanding the olfactory surface. Alistrosaurus, one of the few survivors of the end Permian extinction, could detect the faint scent of green vegetation kilometers away after volcanic winters had blanketed the landscape in ash. Good smells became beacons of recovery. Bad smells, the sulfur and halocarbons from continuing eruptions, signaled areas to avoid. The sense of smell was now mastered in several dimensions. Detection range increased from centimeters to kilometers in favorable wind conditions. Discrimination reached hundreds of molecular classes. Memory allowed animals to form olfactory maps of their world. Social use expanded to include mate choice, parent-offspring recognition, territory advertisement, and alarm signaling. Learning shifted from purely individual trial and error to include observational and even cultural elements within groups. As the Triassic dawned after the Great Extinction, archosaurs and the first true mammals inherited these refined olfactory toolkits. In river valleys of what is now Argentina and southern Brazil, early cynodonts and dicynodonts continued to rely on smell for survival in increasingly seasonal climates. Their descendants would carry the torch forward, eventually passing it to the primate lineage that leads directly to us. Throughout these hundreds of millions of years, the core mechanism remained consistent. Good smells reliably predicted resources critical for survival, food, mates, safety, while bad smells reliably predicted threats, toxins, predators, disease, environmental extremes. Learning began as genetic hardwiring sculpted by natural selection, then layered with individual experience, then with social transmission. Mastery meant turning an invisible chemical landscape into a readable, predictable, navigable map of the world. This aquatic to terrestrial transition, this slow refinement across Paleozoic seas, swamps, and floodplains, created the olfactory hardware and software that early mammals, and eventually humans, would inherit and further elaborate. The stage was set for warm-blooded, nocturnal creatures to make smell their dominant sense in a world of darkness and danger. We now enter the world of the first true mammals and their immediate predecessors, a world shaped by darkness, small body size, and the constant threat of larger predators. This third chapter carries us through the late Triassic, Jurassic, Cretaceous, and into the earliest days of the Paleogene, roughly 230 million years ago to 50 million years ago. During these periods, the sense of smell did not merely survive, it became the dominant sense for an entire major lineage. Mammals turned olfaction into their primary window on reality, refining it far beyond what had existed before. They learned to read incredibly faint traces across vast distances, to remember complex scent landscapes over seasons and years, to use odors as maps, signatures, warnings, and invitations. Mastery reached new heights. Mammals began to manipulate smells deliberately, marking territory, masking their own presence, and even broadcasting information to kin across generations. The story begins in the colorful badlands of what is now the Izquigualasto Formation in northwestern Argentina, around 231 to 225 million years ago. Here, in the humid river valleys of western Pangaea during the Carnian stage of the late Triassic, the very first mammals, or at least their closest cynodont ancestors, lived alongside early dinosaurs. Fossils of tiny, shrew-like forms such as Brachylaudin and Morganucodon show skulls in which the olfactory region occupies an astonishing proportion of the brain case. In some specimens, the olfactory bulbs are larger, relative to total brain size, than in almost any living mammal except certain specialized scent hunters like Solenodons or tenrecs. These creatures were strictly nocturnal, insectivorous, or omnivorous, rarely larger than a modern mouse. Vision was secondary. The moonlit forest floor offered little to see. But the air carried endless streams of information. The musky trail of a passing insect larva, the sharp alarm chemical released by a disturbed millipede, the faint resinous perfume of a ripening pteridosperm cone, the acrid ammonia of a carnivorous archosaurus marking a hunting ground to be avoided. Good smells were literally life. The sweet, protein-rich volatiles leaking from a beetle grub hidden under bark drew these early mammals straight to a high-calorie meal. The earthy, fungal odor of decaying wood after rain signaled colonies of termites or grubs. The delicate floral notes of early Benetidalian flowers promised nectar or pollen. Bad smells were equally precise warnings. The sulfurous reek of fresh theropod scat meant a Herosaurus or similar predator had passed recently. The bitter, alkaloid-laden volatiles from certain ferns and cycads advertised toxicity. The heavy, cloying stench of anaerobic rot in a waterlogged hollow warned of low oxygen and possible ambush sites. Learning was brutally efficient. An individual that ignored a warning scent once might not live to ignore it again. Survivors passed on both their genes, favoring keener receptors and larger olfactory bulbs, and their behavioral tendencies through observation of parents and siblings. By the early Jurassic, around 200 to 175 million years ago, true mammals had appeared. In fissure-filled deposits of what is now Britain, Wales, and Portugal, sites like Pant and St. Bride's, paleontologists have recovered exquisitely preserved jaws and skulls of docodonts, eutriconodonts, and haramidins. These animals already possess the three-bone middle ear that defines mammals, freeing the quadrate and articular bones for hearing. Yet the olfactory system remained disproportionately large. The nasal cavity contained complex turbinate bones, scroll-like structures that dramatically increased the surface area available for olfactory epithelium. Modern reconstructions estimate that some early Jurassic mammals had olfactory surface areas comparable to or exceeding those of living bloodhounds, despite bodies only 10 to 20 centimeters long. This hyperdevelopment allowed detection of parts per trillion odorant concentrations carried on faint air currents. These mammals lived in undergrowth, beneath conifer-dominated forests that stretched across much of Laurasia and Gondwana. A good smell might be the yeasty, fermenting aroma of overripe benetite seeds fallen to the forest floor, or the warm, lipid-rich scent of a clutch of beetle eggs. A bad smell could be the musky territorial marking of a tritylodont-cynodont competitor, the smoky residue of a lightning-caused ground fire, or the coppery blood scent of a recent predation event. Mastery here included active scent manipulation. Many small mammals groomed themselves with saliva containing antimicrobial compounds, reducing their own body odor to avoid detection. Some likely rubbed against strong-smelling plants, resinous conifers, pungent ferns to camouflage themselves, a behavior seen today in hedgehogs and some rodents. Territory marking with urine and anal gland secretions became routine. The lingering scent profile told neighbors' size, sex, reproductive status, and health. A rival reading these chemical billboards might choose to avoid confrontation. The Middle and Late Jurassic, 175 to 145 million years ago, saw mammals diversify while still remaining mostly small and nocturnal. In the famous Daehugu and Yenliao biotas of Liaoning Province, China, exquisitely preserved fossils show fur, external ears, and in some cases even soft tissue outlines of nasal structures. Castorocauda, a semi-aquatic docodont, had nostrils positioned for sniffing while swimming. Volaticodurium, a gliding eutriconodont, relied on smell to locate insect swarms while gliding between trees. Learning became more sophisticated. Juveniles followed their mothers, sent trails back to nests after short forays. Adults remembered seasonal changes in odor landscapes. The sudden burst of monoterpenes when certain conifers released pollen, the damp mushroomy notes after summer rains that signaled mushroom-fruiting bodies full of protein. Bad smell memory was especially long-lasting. A single encounter with the defensive quinone spray of a bombardier beetle could produce lifelong avoidance of similar sharp, irritating volatiles. By the Cretaceous, 145 to 66 million years ago, some mammal lineages had grown larger and more ecologically diverse. In river floodplains of what is now Mongolia and North America, a Catalgod dinosaur provincial park, multituberculates, metatherians, and early eutherians foraged under the feet of tyrannosaurs and ceratopsians. Multituberculates, rodent-like but with completely different teeth, had among the most elaborate turbinates ever recorded in Mesozoic mammals. Their olfactory acuity allowed them to exploit patchily distributed food resources, fungal spores, seeds, insect larvae, small vertebrates. Good smells guided them to windfalls, clusters of cycad seeds split open by beetles, resin droplets trapping nutritious insects. Bad smells steered them away from predator latrines, toxic plant latex, and anoxic swamp pockets. Social olfaction reached new complexity. Some Cretaceous mammals likely lived in small family groups. Mothers recognized pups by individual scent signatures. Alarm odors, sharp carboxylic acids released from anal glands, could scatter a group instantly. Males advertised fitness through strong, complex pheromones secreted from specialized skin glands. Females selected mates partly on olfactory compatibility, a system still seen in mice and many primates today. This chemical mate choice reduced inbreeding and optimized immune system diversity in offspring. The end Cretaceous extinction event 66 million years ago eliminated non-avian dinosaurs but spared most small mammals. In the first few million years of the Paleocene, in places like the Fort Union Formation of Montana and the Shanghulu fauna of China, mammal diversity exploded. Placental and marsupial lineages radiated rapidly. Olfactory dominance persisted and even intensified in many groups. Early primates, plesiadapiforms such as Purgatorius from Saskatchewan, retained enormous olfactory bulbs while beginning to develop better color vision. In the dense, warm forests that covered much of the globe during the Paleocene-Eocene thermal maximum, around 56 million years ago, early euprimates like Tailhardina and Cantius foraged in the understory and lower canopy of what are now Wyoming, Belgium, and Pakistan. Here smell remained critical. The sweet, overripe volatiles of strangler figs ripening high in the canopy drew primates upward. The musky scent of group members helped maintain cohesion in dense foliage. The bitter, tannin-heavy odor of unripe or toxic leaves prompted avoidance. Bad smells included the ammonia-rich urine trails of large terrestrial predators that occasionally passed below, the acrid smoke of natural fires, the putrid gases of anaerobic leaf litter. Learning was now multi-generational and proto-cultural. Infants watched mothers sniff suspicious fruits before tasting. Adolescents learned which scent trails led to reliable water holes during dry seasons. Elders with scarred noses and faded pelage still remembered the distinctive rotten meat odor that had once signaled a saber-toothed predator's cached kill years before. By the Eocene proper, 56 to 34 million years ago, in lush lake and river systems of the Green River Formation, Wyoming, Messel Pit, Germany, and Quercy-Phosphorites, France, primates diversified into lemur-like, tarsier-like, and early anthropoid forms. Darwinius massillii from Messel shows an olfactory bulb still large relative to neocortex. These animals navigated three-dimensional forest worlds where visual cues were often blocked by leaves. Smell provided long-range information, the fruity aroma of a distant fig tree in fruit, the floral perfume of night-blooming vines that promised nectar, the heavy musk of a rival male marking a territory boundary. Bad smells warned of pythons coiled in branches, of toxic millipedes, of stagnant pools harboring disease. Mastery now included deliberate environmental modification. Some early primates chewed strong-smelling resins and rubbed them on fur, possibly for parasite control or social signaling. Others selected sleeping trees with aromatic leaves that repelled biting insects. Group members shared knowledge of good and bad scent patches, localities where certain odors reliably predicted food or danger across years. This olfactory tradition formed one of the earliest substrates for primate culture. Throughout this 180 million year arc, from tiny Triassic insect eaters hiding underfoot to Eocene primates leaping through canopy gaps, the sense of smell evolved from a simple survival detector into a rich, multi-dimensional sense. Mammals learned to detect traces at extraordinarily low concentrations over long distances, discriminate among thousands of molecular variants formed detailed. Long-lasting olfactory maps of home ranges use odors for individual recognition, kin identification, mate choice manipulate their own scent profile for concealment or advertisement transmit olfactory knowledge. Socially across generations, good smells consistently guided toward nutrition, reproduction, safety. Bad smells shielded from poison, predation, disease, conflict. The hardware, vast olfactory epithelium, elaborate turbinates, large olfactory bulbs was in place. The software instinct, individual learning, social tradition was increasingly sophisticated. When the primate lineage that would eventually lead to humans continued its journey through the forests of Africa tens of millions of years later, it carried with it this extraordinarily refined olfactory heritage. The stage was set for the next great transformation, the partial shift from smell-dominated perception to one increasingly shared with vision, yet never abandoning the ancient power of the nose. We now step into the long Miocene and Pliocene epochs, roughly 23 million to 2.6 million years ago, when the primate lineage that would eventually produce our own genus Homo lived almost entirely within the warm, humid, and increasingly seasonal forests and emerging woodlands of Africa, with small extensions into southern Eurasia. This fourth chapter focuses on the profound olfactory life of early apes and the first hominins. During these millions of years, the sense of smell remained a cornerstone of survival, navigation, foraging, social bonding, and danger avoidance. Even as color vision strengthened, hands became more dexterous and brains slowly enlarged. Good and bad odors were not mere background sensations. They constituted a primary geography of the world, a chemical map more detailed and reliable in many contexts than anything the eyes could yet provide. Around 23 to 20 million years ago, in the early Miocene forests that covered much of eastern Africa, sites now exposed in the Tugen Hills of Kenya, Rusinga Island in Lake Victoria, and the Koru and Meswa Bridge localities, the proconsulates flourished. Proconsul, the best-known genus, ranged in size from a small cat to a large dog. Its skull fossils show olfactory bulbs that, while smaller relative to brain size than in Mesozoic mammals, were still very large compared with living Old World monkeys or apes. The nasal cavity housed intricate turbinates, and the cribriform plate, the perforated bone through which olfactory nerves pass, was broad. These early apes moved quadrupedally through multi-layered canopies, feeding on ripe fruit, young leaves, insects, and occasional small vertebrates. For proconsul, smell was indispensable. The heavy, fermenting sweetness of overripe figs bursting on the branch drew individuals from hundreds of meters away, especially on still mornings when thermal updrafts carried volatiles upward. The sharp, green, grassy volatiles released when certain vines were crushed underfoot signaled fresh, tender shoots safe to eat. The delicate, musky floral notes of certain lianas blooming at night guided nocturnal feeding bouts. Good smells were precise locators of calorie-dense, low-toxin food patches in a forest where visual detection was often blocked by foliage. Bad smells were equally precise sentinels. The bitter phenolic odor of unripe or toxic strychnose fruits warned against ingestion. The acrid, quinone-laden defensive spray of certain millipedes encountered on the forest floor triggered instant retreat. The heavy, musky territorial scent deposited by a larger male on a frequently used branch crossing announced dominance and prompted subordinates to choose alternate routes. Learning in these communities was intensely social and developmental. Infants clung to mothers and learned by imitation. When a mother paused to sniff a suspicious fruit, the infant watched the facial expression and body posture that followed. If the mother discarded the fruit with a grimace and head shake, the young associated that particular volatile profile, perhaps a mix of isomyl acetate and certain aldehydes, with rejection. Juveniles followed older siblings to reliable fruiting trees, memorizing not just location, but the characteristic seasonal scent signature that preceded ripeness by days. A youngster that once ate a mildly toxic leaf and suffered hours of nausea carried that aversive memory for life, avoiding similar bitter green odor blends even decades later. Elders, with their accumulated scent catalogs, served as living libraries of what was safe, nutritious, or dangerous. By the Middle Miocene, 17 to 11 million years ago, the climate began to dry in many regions. In the rift valleys of Kenya and Ethiopia, localities such as Maboko Island, Nakola, and the Njernjua beds, apes like Keniapithecus, Equatorius, and Nakalapithecus lived in more open woodlands interspersed with denser gallery forests along rivers. Here the olfactory world shifted subtly but importantly. Forest fruits became patchier, grasslands and seasonal water sources appeared. Good smells now included the faint, sweet grass volatiles carried on dry season winds from distant riverine fig groves. The mineral-rich, slightly saline odor of certain clay licks drew individuals to supplement sodium and other minerals. The warm, yeasty aroma of fallen marula like fruits fermenting on the ground promised a quick energy source during food shortages. Bad smells gained new significance. Dusty, urine-soaked scent of large carnivore latrines, saber-tooths, bear dogs, hyenodonts marked zones to skirt. The acrid smoke of dry season bush fires, carried tens of kilometers, signaled both danger and opportunity. Flee the flames, but later return to the scorched ground for roasted tubers or insects. The putrid ammonia-heavy stench of stagnant water holes warned of bacterial contamination and crocodiles. Olfactory memory became even more critical in these patchy landscapes. An individual that remembered the precise blend of terpenes and esters that preceded peak fruiting in a particular grove could return year after year at exactly the right time, gaining a survival edge. Social olfaction deepened. Males developed prominent sternal and axillary glands, rubbing secretions onto branches and trunks to advertise status, readiness to mate, and group affiliation. Females in estrus released subtle volatile pheromones that drifted through the woodland, allowing distant males to assess fertility without visual contact. Infants were recognized by unique scent signatures blending maternal odor with their own. Group cohesion relied partly on shared familiarity with a collective home-range scent profile, the characteristic mix of plant volatiles, soil odors, and conspecific markings that defined safe territory. Intruders carrying foreign scent blends triggered defensive displays or chases. Around 10 to 7 million years ago in the late Miocene, the last common ancestor of African apes and humans lived in forests and woodlands stretching from what is now Ethiopia through Kenya to Uganda and possibly farther west. Sites such as Ne Kali, Samburu, and Korora preserve fauna suggesting mosaic environments, closed canopy patches, open woodland, and seasonal grassland. The last common ancestor likely resembled a generalized great ape, suspensory locomotion, large body size in males, complex social structure. Its olfactory system was still robust, large olfactory epithelium, elaborate turbinates, prominent bulbs. Smell remained essential for long-range detection in visually cluttered environments. Good smells guided to ephemeral resources. The honey-like aroma of stingless bee nests high in hollow trees, the rich, lipid, heavy scent of oil-rich nuts ripening in scattered palms, the fresh, green volatiles of new grass shoots after early rains. Bad smells protected against an expanding guild of predators, false saber-tooths, large hyenas, pythons, eagles. The coppery blood and fear scent of a recent predation event lingered for days, marking a locality to avoid. The sulfurous, rotten egg odor of geothermal springs in rift valleys warned of toxic gases. Around 7 to 6 million years ago, the human-chimpanzee split occurred, most likely somewhere in the forested rift valleys or adjacent woodlands of Central or Eastern Africa, possibly near present-day Lake Turkana or farther west in the Congo Basin region, though direct fossils remain elusive. The earliest potential hominin lineage, represented by Sahelanthropus tchadensis from Toros Menala in Chad's Jurab Desert, dated to ~76 Ma, already lived in a mosaic of gallery forest, woodland, and grassland. Its foramen magnum position suggests some bipedal capability, yet the cranial base preserves space for a large olfactory region. For these earliest putative hominins, olfaction was still a dominant sense. They foraged in gallery forests along ancient Lake Chad's tributaries. Good smells included the fruity volatiles of wild dates and figs along watercourses, the nutty aroma of oil-rich seeds in open woodland, the mineral scent of termite mounds after rain. Bad smells warned of seasonal anoxia in drying pools, the musky territorial markings of large male crocodiles, the sharp ammonia of big cat latrines on game trails. Bipedalism, even if facultative, raised the head higher, improving air sampling and long-distance scent detection on breezy savanna edges. By 6 to 4 million years ago, Ororan tugenensis from the Tugen Hills and Ardipithecus cadaba from the Middle Awash region lived in similar mosaics. Ardipithecus ramidus, best known from Tilde 4 Ma deposits at Aramis in the Afar region of Ethiopia, provides the clearest window. Ardi's remarkably complete skeleton shows a mix of arboreal and terrestrial adaptations. Cranial fragments and associated fauna indicate close to open woodland with abundant figs, palms, and hackberries. Pollen and isotopic data confirm a diet rich in C3 forest plants and fruits. Ardipithecus relied heavily on smell. The sweet, overripe odor of strangler figs attracted groups to feeding trees. The faint honeydew scent of Homopteran honeydew on leaves guided to insect-rich patches. The musky, glandular odor of group members maintained social bonds during dispersed foraging. Females likely used olfactory cues to assess male quality, complex blends of androgens and health-related volatiles. Males marked resources and pathways with urine and glandular secretions, creating a chemical landscape of ownership and invitation. Bad odors were omnipresent threats. The bitter alkaloid profile of certain unripe or toxic fruits prompted immediate rejection. The putrid stench of decaying carcasses drew scavengers but warned of disease risk. The acrid, musky urine trails of leopards and saber-tooths marked ambush zones to circumvent. During the dry season, the dusty, fecal-heavy odor of distant waterholes carried on hot winds guided groups to reliable sources, while the stench of desiccated, cyanobacteria-choked pools warned to keep moving. Learning was multi-layered. Infants rode on mothers' backs, noses close to maternal fur, imprinting on the safe baseline scent of the group. Juveniles shadowed older individuals, learning which faint trails led to reliable tubers or water seeps. Adults remembered seasonal scent shifts, the sudden burst of monoterpenes when certain acacias flowered, signaling edible gum. A single bad experience, gastrointestinal distress after eating a mildly toxic berry, could produce lifelong avoidance of that precise odor blend. Around 4 to 2.6 million years ago, in the Pliocene, Australopithecus and Kenyanthropus emerged in eastern Africa. Canopoi, Aliabae, Litali, Hador, Waranso, Mill, Dicica. These early hominins were habitual bipeds with ape-sized brains yet increasingly dexterous hands. Their environments ranged from closed woodland to bushland to open grassland with riverine forests. Isotopic studies of teeth show mixed C3 and C4 diets, meaning they exploited both forest fruits and savanna resources. Olfaction remained vital. Good smells guided to patchy, high-quality foods, the fruity volatiles of wild figs and marulas, the yeasty aroma of fallen palm nuts fermenting, the fresh, green scent of young sedge shoots in wetlands. The mineral-rich odor of certain soda lakes drew them to lick for micronutrients. The warm, lipid-heavy scent of scavenged marrow from predator kills offered rare, high-calorie windfalls. Bad smells shielded against diverse dangers. The sharp, carboxylic acid alarm odors released by vervet-like monkeys in nearby trees signaled approaching big cats. The heavy, musky territorial markings of hyenas marked bone-crunching zones to avoid. The ammonia-laden stench of drying urine pools warned of concentrated toxins during drought. Smoke from distant lightning fires carried both warning and promise. Avoid the blaze, but later exploit cooked plant foods left behind. Social olfaction grew ever more complex. Group sizes likely ranged from small family units to multi-male, multi-female bands. Mothers recognized infants by unique scent even in dense vegetation. Males used strong axillary and genital odors to advertise dominance and fertility. Females selected mates partly on olfactory compatibility, favoring males whose immune system-related volatile profiles complemented their own. Groups maintained cohesion through shared familiarity with a collective scent map. The characteristic blend of local plant volatiles, soil odors, water smells, and conspecific markings that defined home. Mastery of smell now included proto-technological elements. Some groups may have carried strong-smelling leaves or resins to mask their own scent while approaching wary prey or avoiding predators. Others selected sleeping trees with aromatic foliage that repelled biting insects, knowledge of which odors reliably predicted seasonal water or fruiting peaks was transmitted across generations, forming the earliest olfactory traditions. By the late Pliocene, around 2.8 to 2.6 million years ago, the first stone tools appeared at Lomqui 3 in West Turkana, Kenya. Whether made by Kenyanthropus, early Australopithecus, or a transitional form, these simple flakes suggest cognitive advances. Yet smell likely still guided raw material prospecting, sniffing for the distinctive earthy mineral odor of high-quality basalt or chert outcrops along riverbeds. Through these 20 million years, from proconsulid apes in closed Miocene forests to bipedal australopiths on Pliocene savanna edges, the sense of smell evolved from a dominant survival sense into a richly layered, socially embedded, culturally transmitted faculty. Good odors reliably led toward nutrition, water, mates, safety. Bad odors shielded from toxins, predators, disease, conflict. Learning shifted ever further from pure instinct toward individual experience, imitation, social observation, and proto-cultural tradition. The hardware, still large olfactory epithelium, intricate turbinates, substantial bulbs, was largely in place. The software olfactory memory, scent-based social signaling, shared environmental knowledge was increasingly sophisticated. When the genus Homo appeared around 2.8 to 2 million years ago, it inherited an olfactory system extraordinarily refined by tens of millions of years of primate evolution in African woodlands and emerging savannas. The next chapter will follow that lineage as brain expansion, tool complexity, fire control, and wider migrations began to reshape how smell was used, even as vision and other senses gained ground. We now arrive at the critical window when the genus Homo first appears and begins its long, intricate dance with expanding brains, stone tools, controlled fire, wider landscapes, and eventually symbolic behavior. This fifth chapter spans roughly 2.8 million years ago to about 300,000 years ago, from the earliest stone tool makers through Homo habilis, Homo rudolfensis, Homo erectus/ergaster, and the first anatomically near-modern Homo sapiens in Africa. During this immense stretch of time, the sense of smell did not diminish in importance. Rather, it was progressively repurposed, layered with new cognitive tools, integrated with emerging technologies, and woven ever more tightly into social, technological, and eventually ritual life. Good and bad odors remained vital guides to survival, but humans began to use them deliberately, to anticipate them through planning, to modify them through action, and in some cases to transform their emotional and symbolic meaning. The opening scene is set in the dry, scrubby lake margins and river floodplains of what is now northern Kenya, around 2.8 to 2.3 million years ago. At Lomqui 3 near the western shore of Lake Turkana, the oldest known stone tools, crude flakes and cores, lie scattered among sediments dated to 3.3 million years ago, though most secure evidence clusters closer to 2.6 million years at sites such as Gona in Ethiopia and Kandjara south in Kenya. Whether these earliest artifacts were made by late Australopithecus, Kenyanthropus, or the first members of Homo, the tool makers lived in mosaic environments, gallery forests along perennial rivers, open acacia woodland, grassy patches, and seasonally flooded lake edges. For these creatures, smell was still the long-range sense par excellence. Vision, while improving with better stereopsis and perhaps rudimentary color discrimination, remained hampered by tall grass, dust, and the low angle of a partially bipedal posture. Hearing carried predator roars and bird alarm calls, but only smell could reveal what lay beyond the next rise or around the river bend hours before arrival. A faint, sweet, acidic blend of fermenting marula or wild fig, carried on the afternoon breeze from kilometers away, could pull a small band toward a windfall feast. The mineral-tinged, slightly brackish odor of a freshwater spring seeping through soda lake sediments guided thirsty groups across parched ground during the dry season. The rich, fatty, marrow-like volatiles drifting from a lion or saber-toothed kill promised high-value scavenging opportunity if the predators had moved on. Conversely, bad smells formed an invisible fence around danger. The sharp, carboxylic acid tang of fresh big cat urine marked a recent territory boundary. Experienced individuals gave those zones a wide berth. The heavy, musky fecal odor of hyena latrines lingered for days, warning of bone-crunching competitors who would contest any carcass. The putrid, hydrogen sulfide-laden stench rising from drying anoxic mud flats signaled bacterial blooms and crocodile-haunted shallows. The acrid, phenolic bite of certain unripe or insect-defended acacia pods prompted immediate rejection even before tasting. Learning accelerated dramatically with the appearance of systematic stone tool use. Tool-making required not only motor skill, but also foresight and memory. Smell likely played a guiding role in raw material prospecting. River cobbles suitable for flaking often carry a distinctive damp mineral odor different from weathered surface stones. Individuals may have learned to seek out those scents along gravel bars after floods. Once a good source was located, its location was remembered not just visually but olfactorily. The characteristic blend of river silt, algae, and sun-baked basalt became a reliable waypoint. Social transmission of olfactory knowledge intensified. A mother who consistently led her offspring to the same grove when its figs reached peak fermentation taught them the precise volatile profile that signaled ripeness days before visual cues were obvious. A scavenger who survived approaching a carcass only after the sharp ammonia scent of big cat urine had faded past that temporal sequencing rule to others. Wait until the warning odor weakens. Infants carried in arms or on backs spent their first years with noses close to parental skin, imprinting on the group's collective baseline scent while simultaneously sampling the world's odors through close contact. By around 2.4 to 1.8 million years ago, Homo habilis and Homo rudolfensis inhabited the same lake basins: Olduvai Gorge in Tanzania, Kubai Fora in Kenya, Omo Kibish in Ethiopia. Brain sizes crept upward to 600–750 cm3. Hands became more precise. Diets broadened to include more meat and marrow, tubers, and hard seeds. Olfaction remained central to foraging ecology. The earthy, starchy volatiles released when digging certain tubers, wild yams, water lily corms, helped locate buried resources invisible from the surface. The faint, lipid-rich scent of brain or marrow inside a herbivore skull guided percussion tool users to smash open crania for fat-rich contents after larger predators had abandoned the kill. Bad smell avoidance grew more nuanced. The coppery metallic blood scent mixed with fear volatiles from a fresh kill indicated active predation. Approaching too soon invited attack. The slower-to-develop putrefaction odors, ethyl mercaptan, cadaverine, signaled safer scavenging windows. Individuals learned to time their approach by the changing scent profile of decay, a chemical clock readable only through the nose. Around 1.9 to 1.8 million years ago, Homo erectus/ergaster appeared, first in eastern Africa, Kubai Fora, D. Manassi in Georgia ~1, 8 Ma, and soon spreading widely. Brain size jumped to 850–1100 cm3. Bodies became taller, limbs longer, pelage reduced. Most dramatically, by at least 1.5 million years ago, and possibly earlier, controlled use of fire entered the behavioral repertoire at sites such as FXJJ20 Site Complex at Kubai Fora, Wonderwork Cave in South Africa, and later Jesher Benat Yaakov in Israel, ~790 Ka. Fire transformed olfaction in profound ways. The first intentional hearths produced smoke, char, and roasted food volatiles never before encountered in such concentration. Good smells multiplied: the caramelized Maillard reaction sweetness of roasted tubers, the savory protein pyrolysis aroma of seared meat, the nutty toasted scent of heated acacia pods. These novel odors became powerful attractors, drawing group members to share food and warmth. The smell of a distant cooking fire carried far on night air, serving as a beacon for stragglers or a signal of occupation to neighboring bands. Yet fire also introduced powerful new bad smells. Green wood smoke is acrid and eye-watering, laden with phenols and aldehydes. Overheated fat produces acrolein, a sharp, choking irritant. Burning toxic plants, certain euphorbias, for example, releases bitter, nauseating fumes. Early fire users quickly learned which woods produced clean, sweet smoke versus thick, bitter clouds. They selected dry hardwoods for nighttime fires and green branches for daytime smudging to repel insects. Mastery of fire thus included mastery of its olfactory consequences. Fire further amplified olfactory learning. A child who burned its hand on coals associated the sharp, singed hair odor with pain for life. Groups remembered which tree species produced the most pleasant smoke for comfort and which created repellent fumes useful against mosquitoes. Fire-altered landscapes changed scent maps. Burned patches smelled of charcoal and ash for months, signaling past human presence, and altered vegetation regrowth patterns rich in tender shoots. Homo erectus carried these skills across continents. By 1.6–1.2 million years ago they reached Java, Trinil, Sangaran. By 1.20–0.8 million years ago Zhaokudian near Beijing. By ~0, 9 million years ago Gesher, Banat Yaakov in the Levant. In each new environment they recalibrated their olfactory dictionaries. In Southeast Asian rainforests the heavy, resinous sweet volatiles of dipterocarp fruits and the musky odor of strangler figs became good smell beacons. The dank, moldy rot of monsoon-soaked leaf litter warned of fungal toxins. In northern China during glacial cycles, the crisp, dry grass volatiles of autumn signaled harvestable seed patches. The sharp, ammonia-heavy freeze-thaw urine scents of cave hyenas marked winter refugia to avoid. Around 800,000–500,000 years ago at sites such as Boxgrove in England, Arago in France, and Bodo in Ethiopia, larger-brained late Homo heidelbergensis, early archaic sapiens-like populations show even more complex behaviors. Symmetrical handaxes imply aesthetic preferences. Wooden spears at Schöningen, Germany, ~300 Ka indicate active hunting. Olfaction supported these advances. Hunters likely trailed game by following faint musk trails over long distances, distinguishing fresh spoor from hours old by subtle shifts in volatile composition. The coppery blood scent on wind helped locate wounded animals. The musky territorial markings of rival carnivores warned of competition. Social life grew denser. Larger group sizes demanded finer individual recognition. Each person's unique blend of sweat volatiles, breath volatiles, skin bacteria byproducts, and environmental odors picked up from habitual ranging areas created a chemical signature as individual as a fingerprint. Mothers recognized lost children by scent alone in dense bush. Mates identified each other after separation. Dominant individuals broadcast status through stronger axillary odors amplified by diet and health. By 700,000–300,000 years ago, in Africa and Eurasia, populations ancestral to Homo sapiens show increasingly modern cranial features. At Jebel Irhoud in Morocco, ~315 Ka, Florisbad in South Africa, ~259 Ka, and Hertu in Ethiopia, ~160 Ka, but with earlier roots, brains approached modern volumes. Symbolic behavior glimmered, ochre use, shell beads, complex hunting. Smell entered symbolic domains. At Pinnacle Point in South Africa, ~164 Ka, and Border Cave, ~274 Ka, people collected and heated silkreed for better flaking and gathered shellfish. The briny, iodine-rich volatiles of tidal zones became associated with reliable protein. Burned ochre and heated shells produced distinctive earthy pyroligneous aromas that may have carried social meaning, perhaps signaling group identity or ritual preparation. The smell of ochre powder ground with fat and applied to skin could have served as a group marker, a precursor to body painting and perfume. In Europe, Neanderthals, close cousins or sister lineage, used fire systematically from tilde 400 ca onward. At Abric Romani in Spain, Kebara in Israel, and Spy in Belgium, they maintained hearth-centered camps. They selected specific plants for bedding, yarrow, chamomile, whose volatiles repelled insects and possibly had medicinal effects. The aromatic smoke of certain herbs thrown on fires created pleasant or ritual atmospheres. At Bruniquel Cave in France, tilde 176 ca deep underground structures suggest symbolic use of space. The lingering scent of torches and human bodies in those confined chambers would have been overpowering, perhaps deliberately managed. Across Africa, early Homo sapiens populations refined olfactory knowledge into near-modern complexity. At Olorgesili in Kenya, tilde 320-50 ca, they transported obsidian over long distances. Smell may have helped locate high-quality outcrops by their distinctive volcanic mineral odor. At Glacial's river mouth in South Africa, tilde 120-60 ca, they exploited shellfish, seals, and plants. The layered midden odors, brine, guano, charred bone, became part of the sensory home. Good smells now included the comforting aroma of a well-tended hearth, the savory promise of roasting meat, the sweet resin scent of medicinal plants deliberately collected. Bad smells remained warnings, spoiled food volatiles signaling bacterial danger, predator urine marking active hunting zones, smoke from uncontrolled wildfires signaling evacuation, but humans increasingly controlled or avoided them through foresight, planning, and group coordination. By 300,000 years ago, the olfactory system of Homo sapiens was anatomically modern. Tilde 400 functional odorant receptor genes, massive piriform cortex integration with memory and emotion centers, tight links to limbic structures governing fear, pleasure, and social bonding. Yet the way smell was used had been transformed by cumulative cultural evolution. Knowledge of which odors predicted seasonal resources, which signaled safe water, which warned of hidden threats was no longer purely individual or even group-specific. It was transmitted across generations through language precursors, demonstration, and shared experience around fires. The stage was now set for the Upper Paleolithic explosion, symbolic culture, art, long-distance trade, ritual burial, and deliberate scent manipulation through resins, flowers, and eventually perfumes. But that acceleration belongs to the next chapter. Here, in the deep time from 2.8 million to 300,000 years ago, smell transitioned from being the dominant window on an uncertain world to a richly integrated sense, still vital for survival, now amplified by fire, tools, foresight, social complexity, and the first glimmers of symbolic meaning. Homo had not lost its nose. It had learned to think with it, live with it, and in some cases begin to shape the world's odors to its own purposes. We now cross into the period when Homo sapiens fully emerged as a species capable of symbolic thought, long-distance social networks, elaborate material culture, and deliberate sensory manipulation. This sixth chapter covers roughly 300,000 years ago to about 40,000 years ago, from the earliest anatomically modern humans in Africa through the African Middle Stone Age, the initial dispersals out of Africa, the European arrival of our species, and the beginning of the Upper Paleolithic cultural explosion. During these hundreds of millennia, the sense of smell was not pushed aside by expanding visual symbolism or language. Instead, it was elevated, refined, weaponized, ritualized, and in many cases made central to new forms of meaning-making. Good smells became instruments of comfort, identity, attraction, healing, and divine communication. Bad smells were transformed into deliberate warnings, repellents, purifiers, and markers of taboo. Olfaction moved decisively from a purely survival sense into one intertwined with emotion, memory, social hierarchy, technology, art, and the earliest stirrings of religious and aesthetic experience. The journey begins in the diverse landscapes of Middle Stone Age Africa. At Jebel Irhoud in Morocco, dated to years ago, Early Homo sapiens already possessed facial features close to the modern average, brains of modern volume, and a toolkit that included heat-treated stone, projectile points, and ochre processing. In the lush woodlands and grasslands around the site, the air carried layered scent information. The sweet, resinous perfume of acacia gums chewed for calories and dental hygiene, the mineral earthy aroma of ochre-rich clay beds deliberately sought for pigment, the briny mineral tang of nearby coastal resources carried inland on wind. These people collected shellfish when they ventured to the Atlantic edge. The iodine-heavy, ozone-like volatiles of tidal zones became associated with rare feasts of protein and fat. Good smells guided sophisticated seasonal movements. The faint honeydew scent of flowering aloes after winter rains signaled nectar-rich patches attracting bees whose hives could be raided with smoke. The warm, yeasty volatiles of fermenting marula fruit in late summer drew bands to predictable windfalls. The crisp, green grass odor of fresh post-fire regrowth lured grazing herds whose tracks could then be followed. Bad smells shaped avoidance and timing. The sharp ammonia bite of fresh leopard urine on acacia trunks marked kill zones to skirt. The slow-building cadaverine and putrescine stench of a week-old carcass indicated safer scavenging. The sulfurous geothermal fumes near rift hot springs warned of toxic gas pockets. Social life was dense and extended. Groups of 2,150 individuals maintained long-term relationships across dozens of kilometers. Each person carried a unique olfactory signature, sweat volatiles modulated by diet, skin bacteria, hormonal state, environmental odors absorbed from habitual ranging areas. Recognition at a distance relied partly on scent carried on wind. A returning hunter could be identified by kin before visual contact. Mothers and infants shared continuous olfactory intimacy. Breast milk volatiles, skin contact, shared sleeping nests rich with group scent. Estrus females released subtle blends of copulins and androstenol-like compounds that influenced male attention and female alliances. Males competing for status amplified their axillary odors through diet, richer meat producing stronger musky notes, and deliberate rubbing against aromatic plants. Around 200,000 years ago, in southern and eastern Africa, Border Cave, Glacial's River, Blombo's Cave, Pinnacle Point, Diepkloof, populations showed accelerating behavioral modernity. They heat-treated silkrete for sharper tools, fashioned bone points, collected ochre in quantity, perforated shells for beads, engraved ochre plaques with geometric patterns. These innovations had olfactory dimensions. Heated silkrete releases a distinctive hot stone mineral scent during napping. Repeated exposure taught nappers which raw materials produce the cleanest fracture and most pleasant working aroma. Ochre grinding with fat or marrow produced rich, earthy, buttery volatiles that clung to skin and clothing, perhaps functioning as a group marker or cosmetic enhancer. Shell beads worn against skin absorbed personal scent over months, becoming olfactory extensions of identity. At Pinnacle Point, 164,000 years ago, people exploited a wide coastal niche. The layered midden smells, brine, charred bone, guano, decaying kelp, created a characteristic home odor profile. When groups moved seasonally inland, they carried dried shellfish whose lingering marine volatiles reminded them of the coast. Smoke from controlled burns shaped the landscape. The sweet smoke of certain hardwoods was preferred for cooking, while acrid green wood smoke repelled insects during wet seasons. Medicinal plants were deliberately burned or crushed. The camphoraceous, menthol-like volatiles of certain Helichrysum species soothed respiratory irritation, creating learned associations between specific aromas and relief. By 120,000 years ago, early dispersals out of Africa are documented. At Eskul and Kafse caves in Israel, burials included red ochre-covered bodies and deer antlers placed near skulls. The ochre, mixed with fat or water, produced a thick, iron-rich scent that permeated the grave fill. These burials suggest that smell played a role in mortuary ritual. The The deliberate application of strongly scented pigment may have masked decomposition odors, honored the dead with a beautiful aroma, or marked transition to another state. Nearby Middle Paleolithic hearths show systematic use of aromatic woods and herbs. The lingering smoke of juniper or pistacia created a distinctive, purifying atmosphere. A second, more sustained dispersal, 50,000 years ago, carried modern humans into South Asia, Southeast Asia, Australia, and eventually Europe. In each new biome, they recalibrated olfactory knowledge with remarkable speed. In the tropical rainforests of what is now Sri Lanka and Borneo, Batadamba Lena, Naya caves, they targeted arboreal game and collected aromatic resins. The heavy, balsamic sweet scent of dammer and copal resins became prized for hafting adhesives, fire starters, and possibly incense-like uses. The musky odor of flying squirrels and colugos guided nocturnal hunts. The sharp defensive stink of certain stink bugs was avoided or used to repel larger predators. In the arid and semi-arid zones of Arabia and the Levant during MIS 5-13080 Ka, and later MIS 43, humans tracked ephemeral water sources by scent. The faint, green mineral volatiles of distant springs carried on night air-guided movements across gravel plains. The dusty, urine-concentrated stench of ibex latrines marked reliable but contested water holes. When groups sheltered in caves during sandstorms, the confined space amplified human body odors, hearth smoke, and plant-bedding volatiles, creating intense, multi-generational scent memories tied to safety. By 45,000 40,000 years ago, modern humans entered Europe, encountering Neanderthals already present for hundreds of thousands of years. In the Danube corridor, Swabian Jura, and later across Western Europe, Aurignacian sites, Jason Klurstrel, Vogelherd, Hohl, Fels, Chauvet, Willendorf, our species brought a dramatically expanded behavioral repertoire: figurative art, musical instruments, elaborate body adornment, long-distance trade, systematic use of scent-bearing materials. Olfaction was central to this cultural leap. At Hohl, Fels, and Vogelherd, perforated bear teeth and fox canines were worn against skin for years, absorbing personal scent and becoming olfactory talismans. Mammoth ivory beads and pendants carried the faint, oily keratin smell of worked ivory that grew richer with handling. Red ochre, transported over hundreds of kilometers, produced a characteristic metallic, earthy aroma when mixed with animal fat for body paint or pigment crayons. Wearing ochre created a shared group scent that persisted through days of hunting and travel. Cave environments amplified olfactory experience. At Chauvet, 33 Ka, the deep chambers smelled of wet limestone, torch soot, human sweat, pine resin torches, and occasional bear musk. Paintings were executed in total darkness save for torchlight. The flickering light and shifting smoke created dynamic scent visual interactions. Bison, lions, rhinos, and mammoths, animals whose musky, fecal, urine, and glandular odors were well known on the surface, were rendered with acute attention to posture and anatomy, suggesting that olfactory memory informed artistic choice. The smell of charcoal and manganese crayons lingered on fingers and transferred to skin, perhaps marking ritual specialists. Aromatic plants were deliberately introduced into living spaces. At Abric Romani and other sites, pollen profiles show yarrow, chamomile, and other medicinals used as bedding. Their crushed volatiles repelled parasites and created a soothing sleep environment. At Grotte du Renne and other Chalopyrrhonian layers, possibly Neanderthal but contemporaneous similar patterns appear. Thrown on hearths, these herbs produced clouds of camphor, cineol, and thymol-rich smoke, pleasant, cleansing, possibly medicinal or ritual. Personal adornment reached new heights. Mammoth ivory beads, perforated shells, eagle talons, fox teeth were strung on cordage and worn against pulse points where sweat volatiles mixed with material odors. Over time, each ornament became impregnated with the wearer's unique chemical signature, functioning as portable identity markers. When exchanged in trade networks spanning hundreds of kilometers, these scented objects carried olfactory proof of distant alliances. Hunting strategies relied on sophisticated olfactory tracking. Aurignacian hunters pursued reindeer, horse, mammoth across open steppes and wooded valleys. The musky trail of a bull mammoth in must could be followed for days. Experienced trackers distinguished freshness by subtle shifts in volatile intensity and composition. The coppery blood scent on wind helped locate wounded animals. The sharp alarm volatiles released by herds under stress guided ambushes. Burial practices incorporated scent deliberately. At Sungir near Vladimir, Russia, 430 Ka, 2 adolescents were buried with thousands of mammoth ivory beads, fox teeth, red ochre, and crucially lances tipped with mammoth ivory. The heavy, oily scent of worked ivory saturated the grave fill. Red ochre, liberally sprinkled, produced a thick iron mineral aroma that would have masked early decomposition and perhaps symbolized vitality or transformation. The entire burial assemblage suggests an intention to surround the dead with powerful or beautiful smells. Across the African savanna, European tundra steppe, Asian forests, and Australian arid zones, humans now used smell as never before, as identity. Personal scent plus adornment plus ochre plus traded materials created individual and group olfactory signatures as medicine. Aromatic plants burned, crushed, or infused soothed pain, repelled insects, aided breathing as ritual. Incense like resins, scented bedding, ochre body mixtures, smoke purifiers marked transitions, birth, initiation, death as technology. Resins for hafting, aromatic woods for preferred fire qualities, scented bedding for parasite control as art. Scent memory informed depiction of animals. Torch smoke and cave volatiles shaped sensory experience of painted chambers as long-distance connection. Scented trade goods carried olfactory proof of relationships across landscapes. Good smells now evoked not only food and safety, but comfort, beauty, memory, status, spiritual power. Bad smells were not merely avoided. They were harnessed, rotten odors used to repel animals, acrid smoke to drive insects, decomposition scents timed for scavenging windows, pungent defensive plant volatiles weaponized against rivals or predators. By 40,000 years ago, the olfactory world of Homo sapiens had become a fully cultural domain. The nose remained a survival instrument, but it had also become a maker of meaning, a shaper of emotion, a carrier of tradition, and a participant in the symbolic revolution that defines our species. The next chapter will follow this scented thread into the full flowering of Upper Paleolithic art, ritual, and social complexity, and onward through the dramatic environmental shifts of the last glacial maximum, where smell once again became a lifeline in increasingly harsh and unpredictable worlds. We now move deeper into the Upper Paleolithic and the turbulent centuries that bridge the height of the last glacial maximum through the warming world of the late Glacial and early Holocene, roughly 40,000 years ago to about 10,000 years ago. In this 7th chapter, the sense of smell reaches one of its most intricate and culturally saturated expressions before the rise of settled villages and agriculture. Across the cold steppes of Europe, the mammoth steppe of northern Asia, the refugia of the Mediterranean rim, the emerging woodlands of the Near East, and the vast continent of Australia already long inhabited, Homo sapiens turned olfaction into an active partner in art, ritual hunting magic, shamanic practice, social signaling, technological ingenuity, emotional regulation, and the first tentative steps toward managing the odors of the dead and the divine. Good smells were deliberately summoned to heal, to attract game, to please spirits, to mark sacred time and space. Bad smells were not only feared and avoided, they were harnessed as weapons, purifiers, tests of courage, boundary markers between worlds, and signals of transformation. Picture the limestone hills of the Swabian Jura in southwestern Germany around 42,000 to 35,000 years ago. At sites such as Hoel Fells, Vogelherd, and Jason Klurstrel, small bands of Aurignacian people sheltered in karst caves overlooking valleys where reindeer, horse, mammoth, woolly rhinoceros, and bison moved in seasonal herds. The air inside these caves was perpetually cool and damp, heavy with the mineral scent of dissolving calcite, the sooty residue of pine resin torches, the musky undercurrent of human bodies living in close quarters for weeks at a time, and the faint, lingering perfume of deliberately introduced plants. Pollen and macrofossil evidence shows that people carried in bunches of yarrow, mugwort, birch leaves, and other aromatics to layer as bedding on the cave floor. When crushed under sleeping hides, these released clouds of sesquiterpenes, cineol, and bitter green volatiles that repelled fleas, ticks, and mosquitoes, while creating a characteristic soothing aroma that generations came to associate with safety and rest. Outside, on the open mammoth steppe, smell was the long-distance sense without rival. Vision could spot herds kilometers away on clear days, but only the nose could read the invisible chemical trails left by animals moving through snow, across rivers, or behind low ridges. Experienced hunters could follow the faint, warm, oily musk rising from the thick winter pelage of a female mammoth in estrus, distinguishing it from the sharper, testosterone-heavy musk of a bull in musth. They learned to read the changing intensity and quality of that musk as wind direction shifted or as hours passed since the animal had passed, fresher notes of intact steroids versus the slow oxidation into stale, rancid undertones. The same subtlety applied to predators. The acrid ammonia and sulfur blend of fresh cave lion urine sprayed on a boulder carried a different temporal signature than the same blend diluted and half frozen after 2 days. Knowing exactly when a marking had been made determined whether a valley was safe to cross or better avoided. Hunting itself became olfactory theater. At sites across Central and Eastern Europe, Dolny Vestanis, Pavlov, Pridmosti, people constructed elaborate traps and drive lanes using scent as lure and barrier. They burned specific woods and herbs to create smoke plumes carrying attractive volatiles toward game trails. The sweet, resinous smoke of burning juniper or pine cones mimicked the scent of certain ripening berries or fresh resin flows that mammoths and reindeer sought in late winter. Other fires were fed damp moss or certain fungi to produce thick, bitter smoke walls that funneled herds into kill zones. The smell of these controlled burns became part of seasonal ritual. When the first juniper smoke rose in spring, it signaled the beginning of the calving season and the return of fat-rich prey. Inside living spaces, scent was curated with care. Hearths were fed preferentially with woods that produced clean, sweet smoke, downy birch, Scots pine, alder, rather than the choking phenol clouds of green oak or spruce. Animal fats dropped onto hot stones sizzled into rich, savory Maillard aromas that filled the shelter and drew people together around the fire. Bone grease, boiled and rendered, carried a warm, nutty perfume that clung to skin and clothing for days, perhaps functioning as an informal group scent marker. When people chewed birch tar pitch obtained by dry distilling birch bark, they released a characteristic phenolic balsamic aroma that marked the mouth and breath of whoever had recently worked adhesives or chewed the tar for pleasure and mild antiseptic effect. Personal adornment saturated the body with scent. At Sungir, burials show adolescents covered in thousands of mammoth ivory beads sewn onto clothing, each bead having absorbed skin oils, sweat volatiles, hearth smoke, and environmental odors over months or years of wear. Fox canine pendants, perforated bare teeth, and discs of mammoth ivory hung against the chest and neck where pulse points released body volatiles that slowly impregnated the ornament. When these objects were traded across hundreds of kilometers, as isotopic and raw material studies confirm, they carried not only visual but olfactory proof of distant connections. A necklace arriving from the Danube basin might still hold faint traces of the limestone dust, pine resin, and human scent of its original wearer. Artistic expression leaned heavily on the nose. The famous Venus figurines of Hoel, Fels, and Willendorf were carved from mammoth ivory that retained its faint, oily keratin smell even after polishing. When held close to the face during carving or during ritual handling, the scent of warm ivory mingled with the carver's breath and hand sweat, creating an intimate sensory bond between maker, object, and viewer. In Chauvet Cave, deep in the Ardèche Valley of southern France around 37,033,500 years ago, the painted panels were executed by torchlight in chambers where the air was thick with limestone moisture, charcoal soot, human perspiration, pine torch resin, and occasional bear musk from hibernating animals. The flickering light made the animals, lions, rhinos, mammoths, horses, appear to breathe. The layered smells made them feel alive. The painters likely chose which panels to work on partly by the acoustic and olfactory character of each chamber. Some spaces smelled sharper and drier, others damper and fungal. The choice of torch wood, pine for bright light and sweet smoke, yew for slower burn, altered the olfactory atmosphere of each painting session. Shamanic or ritual specialists almost certainly used smell to enter and signal altered states. At sites across Europe and Siberia, small quantities of plants containing volatile psychoactive compounds, wormwood, bog myrtle, certain mushrooms, appear in hearth contexts. When burned or ingested, they released pungent, camphoraceous, or musky fumes that could induce visions or heighten sensory acuity. The smell of these substances became a reliable trigger for trance. The first whiff of artemisia smoke rising from coals could shift attention inward, preparing the mind for journeying. In the cold of the Gravettian period, 22,000 years ago, when groups huddled in skin tents or rock shelters during blizzards, these aromatic fires were lifelines, both literally repelling insects in summer, warming in winter, and spiritually. The last glacial maximum, 519,000 years ago, forced populations into refugia, coastal Iberia, the Italian peninsula, the Black Sea littoral, the Franco-Cantabrian region, pockets of the Near East. In these compressed landscapes, smell became even more critical. Resources were patchy. Long-range scent detection could mean the difference between starvation and survival. The salty mineral tang of exposed coastal shellfish beds carried inland on sea breezes guided bands to protein windfalls. The faint, green volatiles of sheltered microclimates where evergreen shrubs persisted through winter storms promised medicinal leaves and kindling. The heavy, ammonia-rich stench of concentrated ibex or chamois latrines marked reliable but overcrowded water sources to approach cautiously. Solutrean and Magdalenian peoples, 12,000 years ago, responded with exquisite technological and artistic refinement. They heat-treated flint, producing a characteristic hot stone smell during napping that experienced flint nappers used to judge temperature and quality. They invented eyed needles of bone and ivory, allowing tailored clothing that trapped body heat and body scent close to the skin, intensifying personal olfactory signatures. They sculpted reindeer antler spear throwers in the form of horses, ibex, and birds. The worked antler retained a faint, horny, marrow-like odor that mingled with the hunter's sweat during use, perhaps believed to transfer the animal's essence to the projectile. In the Magdalenian caves of the Pyrenees and Cantabria, Altamira, El Castillo, Tito Bustillo, Nyaux, deep galleries were used for repeated ritual visits. Torches of birch bark or pine resin left sooty deposits and sweet-smelling smoke trails. Footprints, hand stencils, and engravings were made in near darkness. The smell of wet clay, charcoal dust, human breath, and torch smoke created a multisensory cocoon. Returning to the same panel years later, a person could recognize their own past work partly by the lingering personal scent absorbed into the porous limestone. As the ice sheets retreated after, years ago, forests advanced. In the Near East, Natufian sites in the Levant around 11,500 years ago, people began intensive wild cereal harvesting and gazelle hunting in oak pistachio woodlands. They built semi-permanent structures with plastered floors and collected aromatic plants in quantity, mints, sages, thymes, and wormwood. These were used as bedding, strewn on floors, hung to dry, burned as incense. The characteristic herbal minty camphoraceous aroma of crushed salvia and thymus became the baseline scent of human habitation. When groups returned seasonally to the same site, the smell of old fires, dried herbs, and accumulated human scent welcomed them like a memory made tangible. Burial customs grew more elaborate. At Shanidar Cave in Iraqi Kurdistan, tilde 35,000 years ago, but with later Natufian-like layers, pollen clouds suggest flowers, yarrow, muscary, groundsel were placed with the dead. Even if the famous flower burial interpretation is debated, the deliberate introduction of strongly scented plants into graves is clear in later Natufian and early Neolithic contexts. The sweet, honey-like volatiles of certain composites or the bitter green notes of wormwood masked early decomposition while signaling care, respect, and perhaps an attempt to guide the spirit with pleasant aroma. In Australia, where humans had arrived years ago, long isolated populations developed their own olfactory traditions. In the rock shelters of Arnhem Land and the Kimberley, ochre was ground with animal fat and plant resins, producing thick, metallic sweet pastes applied to bodies and objects. Certain eucalyptus species were burned to produce smoke believed to carry messages to ancestors or cleanse living spaces. The sharp, medicinal cineole scent of eucalyptus became synonymous with healing and spiritual protection. Across all these regions, steppe, tundra, forest edge, Mediterranean scrub, coastal refugia, olfaction had become a fully cultural technology. Hunters read and manipulated animal scent trails with precision, rivaling modern tracking dogs. Ritual specialists curated aromatic atmospheres to induce trance, signal transition, communicate with spirits. Artists and makers chose materials partly for their smell: ivory, antler, resin, ochre. Integrating scent into the aesthetic experience, groups marked identity and territory with shared scent signatures. Body odor plus adornment plus hearth smoke plus chosen plants. Medicine relied on volatile compounds inhaled, absorbed, or burned for pain relief. Parasite control. Respiratory aid. The dead were surrounded with pleasant or symbolically potent aromas to ease passage or honor memory. Good smells evoked nourishment, warmth, community, beauty, spiritual presence, healing, sexual attraction, ancestral continuity. Bad smells, while still warnings of rot, predators, toxins, were now actively managed. Masked with flowers and resins in burials, used as insect repellents, deployed in smoke screens during hunts, tolerated as tests of endurance in initiation rites, or interpreted as omens when spirits were displeased. By the end of this long transition, around 10,000 years ago, as the Holocene dawned, the nose had become not merely a detector but a cultural organ. It read the environment, shaped emotion, carried memory across generations, participated in art and ritual, and helped define the boundary between human and non-human, living and dead, ordinary and sacred. When the first permanent villages appeared and plants and animals began to be domesticated, smell would follow that revolution too, entering granaries, animal pens, cooking hearths, brewing pits, and early metallurgy with entirely new layers of meaning and control. But that agricultural transformation belongs to the next great chapter in the story of how humanity learned to live with, through, and ultimately to reshape the invisible world of odors. We now step across one of the most decisive thresholds in human history, the transition from mobile foragers to sedentary farmers, from wild landscapes shaped by wind and season to human-shaped fields, pens, houses, and villages. This 8th chapter covers roughly 12,000 years ago to about 4,000 years ago, from the very first experiments with cultivation in the Levant and adjacent regions, through the full establishment of Neolithic farming communities across Southwest Asia, Europe, North Africa, the Nile Valley, the Indus region, the Yellow River Basin, and parts of the Americas, all the way to the threshold of the Bronze Age. During these 8 millennia, the invisible world of odors exploded in variety, density, and cultural significance. Smell became not only a detector of the environment but a central architect of the built environment, a marker of social identity, a tool of economic exchange, a participant in new technologies, a language of ritual and cosmology, and for the first time on a large scale, a problem that had to be actively managed, masked, purified, or celebrated in daily life. The story opens in the lush oak pistachio parklands and river valleys of the Levant around 12,000 to 10,500 years ago, during what archaeologists call the early Natufian and then the pre-pottery Neolithic PPNA. At sites such as Ain Malaha, Einan, Wadi Hameh 27, Netiv Hagdud, Gilgal I, and Jericho, people began living year-round in semi-subterranean houses with plastered floors, stone foundations, and storage pits. They harvested wild cereals, barley, emmer wheat in quantity, collected almonds, pistachios, figs, and gazelle in such abundance that some groups could stay put through seasons that once forced movement. With permanence came an intensification of scent. The baseline odor of habitation changed dramatically. Inside circular houses, the air grew thick with the dusty grain sweetness of threshed cereals stored in plastered bins, the warm nutty volatiles of parched barley and roasted pistachios, the smoky resinous smell of fires fed with oak, terebinth, and pistachio wood. People slept on reed mats or goat hair bedding layered with aromatic herbs, thyme, oregano, sage, mint, whose camphoraceous and menthol-like notes drifted upward at night. These plants were not chosen only for comfort. Many carried insect repellent and mild antimicrobial properties. The sharp, cineole scent of crushed salvia leaves became as much a part of home as the sight of the hearth. Storage itself transformed olfaction. Large bins and plastered silos trapped the volatiles released by slowly respiring grains. Over months, a characteristic sweet fermenting, slightly musty aroma developed, pleasant at first, then increasingly sour as molds and weevils took hold if conditions were too damp. People learned to monitor these odors closely. A faint lactic tang signaled acceptable controlled fermentation, early beer-like beverages. A sharp vinegar bite or heavy mold stench warned that the entire season's harvest was at risk. This olfactory vigilance was one of the first systematic human technologies of scent management applied to food security. Around 10,500 to 9,000 years ago, during the pre-pottery Neolithic PPNB, domestication of plants and animals became unmistakable. At Ain Ghazal in Jordan, Sayernu and Nevali Sari in southeast Turkey, Abu Hurira in Syria, and Göbekli Tepe nearby, communities grew visibly larger, some settlements reaching several hundred people. Sheep and goats were penned close to houses. Their musky lanolin-rich fleece odor, urine ammonia, and fecal earthiness saturated the village air. People learned to distinguish healthy animals by the clean, oily smell of their wool versus the sour, metallic tang of sickness or parasite infestation. Milking, initially sporadic, added a new warm, sweet note when fresh milk was brought indoors. Cereal fields expanded. Walking out from the village, one passed through waves of scent, the green grassy volatiles of young barley shoots in spring, the heavy pollen-laden sweetness of flowering wheat in late spring, the dry straw dustiness of ripening heads in summer, the sharp stubble and chaff smell after harvest. These seasonal scent gradients became calendars. When the first honey-like sweetness of flowering figs drifted from the riverine groves, it signaled time to prepare sickles. When the dusty dryness of cut stalks dominated, it was time for thrashing and winnowing. Ritual spaces amplified olfactory symbolism. At Göbekli Tepe, monumental T-shaped pillars, carved with animals, were erected in roofless enclosures around 9680–800 BCE. Before or during gatherings, people burned aromatic plants, juniper, terebinth resin, perhaps wild rue, creating clouds of balsamic piney smoke that rose among the pillars. The smell of burning resin clinging to stone became inseparable from the experience of the place. Animal bones, especially gazelle and aurochs, were deposited in fills. Their slow decomposition added a heavy, iron-tinged putrefaction that may have been deliberately tolerated as part of mortuary or commemorative rites. In some PPNB houses, plastered skulls were mottled with clay and ochre. When freshly prepared, these carried the mineral earthy scent of wet plaster and red pigment, perhaps intended to replace or mask the odors of death. Burial customs grew complex. Under house floors or in special cemeteries, people interred the dead with ochre, beads, and occasionally aromatic plant material. At Tel Halula and other sites, pollen profiles show traces of mint family plants and composites in graves, sweet herbal volatiles that would have softened the early stages of decomposition and perhaps symbolized renewal or safe passage. The deliberate contrast between the heavy putridity of a fresh burial and the cleansing herbal smoke of post-burial rites marked a new olfactory grammar of life and afterlife. As farming spread westward into Europe, linear band ceramic culture till 5540–900 BCE, and eastward into Anatolia, the Zagros, and eventually the Indus Valley and Yellow River regions, each new environment forced a recalibration of the olfactory dictionary. In the less plains of Central Europe, people built longhouses of timber and daub. Inside, the air held the sweet, sappy smell of freshly cut oak beams slowly drying, the earthy mineral scent of daub plaster, the smoky sweetness of beech and hazel hearth fires, the lactic sour notes of stored milk in pottery, the yeasty fermenting aroma of early bread dough rising in clay ovens. Livestock lived in byres attached to or under the same roof. The mingled odors of cattle breath, manure, urine-soaked bedding, and fresh milk created a dense, animal-rich atmosphere that town dwellers today would find overwhelming, but which Neolithic farmers read as signs of prosperity. In the dry farming zones of the Zagros and eastern Fertile Crescent, goat herding intensified. Penning animals near houses concentrated their scents. Musky buck urine during rutting season became so strong it could be smelled hundreds of meters downwind, announcing breeding time to human observers as clearly as it did to other goats. Dairy processing emerged. Fresh milk, souring curds, clarified butter, ghee-like, and fermented products added a palette of lactic, butyric, and cheesy volatiles never before so prominent in human settlements. Pottery revolutionized smell. Fired clay vessels stored liquids and grains without leakage, but they also trapped and concentrated volatiles. Olive oil, sesame oil, linseed oil absorbed into porous fabrics or early pottery surfaces released warm, nutty, fatty aromas when heated. Beer brewing, evident from residue analysis at Godin Teep, Reykjavik Cave, and other sites, produced intense yeasty, malty, sometimes phenolic scents that filled houses during fermentation. The smell of bubbling mash became a seasonal marker of abundance. In the Nile Valley, where farming began till 6000–5000 BCE, the annual inundation created a different olfactory rhythm. Before the flood, the dry fields smelled of cracked earth and old stubble. After the waters receded, the fresh silt carried a rich, mineral green scent of renewal. Emmer wheat, barley, flax, and later emmer spelt fields released waves of pollen and green volatiles. Papyrus marshes added a sweet reedy note, lotus ponds a delicate floral perfume. Mud brick houses trapped cooking smells, roasted fish, garlic, onion, coriander, cumin, whose pungent sweet blend became the signature aroma of village life. By the Chalcolithic period, till 4530–500 BCE, metallurgy added new odors. Copper smelting in the Timna Valley, Fainan, and the Balkans released acrid, sulfurous fumes of roasting ore. Successful smiths learned to judge furnace conditions partly by smell, sweet reducing smoke versus choking sulfur clouds. Arsenical copper alloys produced sharper, metallic volatiles during casting. Beeswax, used for lost wax casting, filled workshops with honeyed sweetness that contrasted dramatically with the metallic bite. In South Asia, the Mayagar sequence, 2500 BCE, shows early cotton cultivation, zebu cattle, and barley wheat farming. Houses of mud brick and plaster accumulated layers of scent, the warm, oily aroma of clarified butter lamps, the smoky resin of Boswellia and Commiphora burned in small braziers, the musky sour smell of penned cattle, the dusty sweetness of stored cotton seeds. In East Asia along the Yellow River, millet farmers of the Yangshao and Dongku cultures, till 5000–3000 BCE, lived in semi-subterranean round houses. Inside, the air held the nutty toasted volatiles of roasted millet, the sour fermenting notes of millet beer, the earthy mineral smell of less plastered walls, the smoky sweetness of broomcorn millet stalks used as fuel. Across all these regions, deliberate scent manipulation became routine. Aromatic plants, mint, thyme, sage, juniper, myrtle, rue, were grown near houses, hung to dry, strewn on floors, burned as incense, used in medicines. Animal products, tallow, lard, milk, cheese, dung, were processed in ways that concentrated characteristic volatiles. Some were pleasant, fresh butter, others pungent, aged cheese, fermenting dung, and tolerated as signs of wealth. Resins, gums, and tars, pistacia, pine, frankincense precursors, were collected, burned, or mixed into adhesives and salves. Early perfumes appear. Fatty bases mixed with crushed flowers, resins, or herbs applied to skin or clothing. Burials used aromatic plants, ochre, resins to mask decay and signal care. Ritual spaces maintained distinctive smoke profiles, sweet for invocation, acrid for purification or exorcism. Good smells now carried layered meanings. Ripe grain equals security, fresh milk equals prosperity, aromatic smoke equals divine presence, personal herbal perfume equals status or attractiveness, clean animal pens equals good husbandry. Bad smells were not merely threats but problems to solve. Spoilage volatiles signaled economic loss. Concentrated manure required removal or composting. Smoke management prevented respiratory harm. Decomposition in settlements demanded burial practices or charnel structures. By around 4,000 years ago, on the eve of the Bronze Age, humanity had created odor-rich artificial ecosystems, villages, towns, early cities, where smell was no longer just read from nature but actively shaped by architecture, economy, technology, ritual, and social hierarchy. The nose had become a cultural instrument as important as the hand or the tongue. When the first cities rose and writing appeared, this scented world would be further codified, traded over long distances, and woven into myth, medicine, and religion with unprecedented sophistication. But that urban revolution belongs to the next chapter in the long story of how humans learned not only to sense good and bad smells, but to create, control, and give them meaning. We now enter the age of cities, writing, long-distance trade, monumental architecture, organized religion, and the first specialized crafts dedicated to scent itself. This ninth chapter stretches from roughly 4000 BCE to about 500 BCE, from the rise of the earliest urban civilizations in Mesopotamia, Egypt, the Indus Valley, and the Yellow River Basin, through the mature phases of Bronze Age societies, the collapse and recovery periods that followed, and into the early Iron Age worlds of the Eastern Mediterranean, Near East, Persia, India, and China. In these 3.5 millennia, the relationship between humans and odor reached unprecedented levels of sophistication, commodification, ritualization, medical application, social stratification, artistic expression, and economic importance. Smell was no longer merely lived with or managed in the home and field. It became a deliberately manufactured, traded, taxed, mythologized, and weaponized dimension of civilization. In the sun-baked alluvium of southern Mesopotamia, where the first true cities, Uruk, Eridu, Lagash, rose between 4000 and 3000 BCE, the air of urban life was saturated with an intensity and diversity of odors never before experienced in such concentration. Narrow lanes between mud brick houses funneled cooking smells, roasted barley bread, garlic, onion, leek, coriander, cumin, dill, into a thick, savory haze that hung over every quarter at mealtimes. From open courtyards came the warm, fatty scent of sesame oil heated in clay pans, the sour fermenting tang of barley beer mash bubbling in large vats, the sweet date syrup aroma of confectionery being prepared for festivals. Sheep and goats were penned in household compounds or driven through streets to market. Their lanolin-rich fleece, ammonia-heavy urine, and pellet-like dung added a constant animal undercurrent. Tanneries along canal edges soaked hides in lime and urine, releasing sharp ammonia clouds that drifted across neighborhoods. Pottery kilns belched smoke laced with the phenolic bite of burning reeds and dung fuel. Copper and arsenic smelting in specialized workshops produced acrid metallic sulfur fumes that stung the eyes and throat. Yet within this dense olfactory texture, certain smells were deliberately elevated and controlled. Temples, the towering ziggurats of Anu, Inanna, Enki maintained their own olfactory identity. Priests burned juniper, cedar, cypress, and myrrh-like resins imported from the mountains of Lebanon and the Arabian Peninsula. The thick, balsamic, piney smoke rose in white columns from braziers on the temple terraces, visible for kilometers across the plain, and carrying a clean, sacred perfume that contrasted sharply with the earthy human-animal odor of the city below. This scent was understood as the breath of the gods. Inhaling it during ritual was to participate in divine presence. When kings processed to the temple gates, they were anointed with aromatic oils, sesame or olive base blended with myrrh, frankincense precursors, galbanum and styrax, whose heavy, sweet, resinous volatiles announced royal divinity and purity. Writing itself captured olfactory knowledge. Cuneiform tablets from the Ur-III period, 2100–2000 BC, list recipes for aromatic substances for the table of the gods, detailing exact quantities of resins, oils, spices, and flowers to be compounded into temple incense. Another genre records perfumes for the king, specifying blends that included imported lapis lazuli dust for color and perhaps symbolic potency, cedar oil, and crushed blossoms. Scribes kept tallies of incoming shipments, boatloads of Dilmun, Bahrain, copper, Magan, Oman, diorite, Meluha, Indus, carnelian, and alongside them, sacks of frankincense, myrrh, cassia, and cinnamon bark from distant lands. Scent materials became prestige goods on par with metals and gemstones. In Egypt of the Old, Middle, and New Kingdoms, 3110–70 BCE, the Nile's seasonal rhythm orchestrated a parallel olfactory civilization. Before the inundation, the dry fields smelled of cracked mud and old stubble. After the flood receded, fresh black silt released a rich, fertile, almost metallic green aroma of renewal. Lotus ponds near temples exhaled delicate floral sweetness at dawn. Papyrus marshes added a green, reedy note. In temple precincts, Karnak, Luxor, Abydos, priests performed daily offerings of incense. Kaiphai, a famous compound of 16 or more ingredients including raisins, wine, honey, myrrh, frankincense, juniper, mastic, cinnamon, cardamom, calamus, rush, and various flowers. When burned on charcoal, Kaiphai released a complex, intoxicating cloud, sweet, balsamic, spicy, floral, that filled hypostyle halls and drifted across the river at sunrise. The scent was believed to awaken the god, nourish the ka, and banish chaotic forces. Workers in the Valley of the Kings inhaled the same smoke during tomb preparation, perhaps believing it protected them from the underworld's dangers. Egyptian perfumery reached industrial scale. Workshops at Memphis and Thebes produced perfumed oils and unguents in huge quantities. Cones of scented fat were worn on wigs during banquets. As body heat melted, the fat, myrrh, lotus, cinnamon, and henna volatiles dripped down the neck and shoulders, creating a slow-release personal perfume cloud. Tomb paintings show servants presenting jars labeled Best Myrrh Oil of Punt or Fragrant Lotus Unguent. Expeditions to Punt, modern Eritrea, Somalia, returned with live myrrh and frankincense trees, saplings planted in temple gardens so the gods could smell their own homeland. In the Indus Valley Civilization, 2600–1900 BCE, Harappa, Mohenjo-Daro, Dhalavira, Lothal cities were laid out on grids with sophisticated drainage that reduced stagnant odors compared with contemporary Mesopotamia. Covered drains carried wastewater away. Public baths, the Great Bath at Mohenjo-Daro, were regularly cleaned, yet scent remained central. Cotton was cultivated and woven into fine textiles that absorbed personal and environmental odors differently than wool or linen. People chewed betel nut and areca, releasing a spicy, peppery tannic aroma from the mouth. Workshops produced carnelian, steatite, and shell beads. Some were strung with aromatic woods or resins. Brick kilns used dung and wood fuel, producing a characteristic earthy, smoky background. Long-distance trade brought lapis from Afghanistan, turquoise from Iran, and most importantly for olfaction, possible early supplies of cinnamon, pepper, and cardamom from South India or Sri Lanka. The famous priest-king statue wears a cloak with trefoil motifs that may represent scented plants. Microscopic analysis has found traces of red pigments possibly mixed with aromatic bases. In East Asia, the Erlitou and early Shang periods, till 1900–1250 BCE, along the Yellow River saw bronze casting workshops that filled the air with the sharp, metallic charcoal smell of molten alloy and burning charcoal. Oracle bone inscriptions record offerings of aromatic grasses and animal fats burned during divination. The resulting smoke was read for omens. Millet beer fermented in large vats released intense yeasty malty volatiles that marked feasting periods. Elite burials included jade, bronze, and lacquered wood. Lacquer sap gave off a distinctive sharp phenolic odor during application, becoming associated with high-status objects. By the late Bronze Age, 1600–1200 BCE, international trade networks, the tin roads, amber roots, incense roots, moved scent materials thousands of kilometers. Mycenaean palaces in Greece imported cedar oil from the Levant, perfumed olive oil in stirrup jars to Egypt and the Hittites. Ugaritic texts list tribute payments in myrrh and frankincense. When the Bronze Age collapse disrupted these networks, 1200–1100 BCE, local substitution became creative. Aegean peoples burned local pine and sage instead of imported resins. Egyptian priests stretched Kai-Fi recipes with Nile lotus and local herbs. In the early Iron Age, 1200–500 BCE, new powers arose. In the Levant, Phoenician cities Tyre, Sidon, Byblos became manufacturing and shipping hubs for purple dye from murex snails, whose rotting flesh produced an infamous stench before yielding the prized Tyrian purple and glass. They also exported cedar oil, galbanum, and storax. The Hebrew Bible describes the tabernacle and first temple rituals with precise incense recipes: frankincense, galbanum, onycha, stacte, burned morning and evening on the golden altar. The smoke was a pleasing odor to the Lord, a technical term that appears dozens of times, indicating a culturally codified olfactory theology. In Persia under the Achaemenids, 550–330 BCE, royal gardens Paradisa grew roses, jasmine, saffron, and narcissus. Their combined floral volatiles were captured in rose water and attar-like distillates. Persepolis reliefs show tribute bearers carrying jars of aromatic oils. Zoroastrian fire temples maintained eternal flames fed with sandalwood and other sweet-smelling woods. The smoke purified the air and carried prayers upward. In India during the Vedic period, 1500–500 BCE, Soma rituals involved pressing a plant, identity still debated, whose expressed juice released pungent, perhaps hallucinogenic volatiles. Agni, the fire god, consumed ghee and aromatic woods. The resulting smoke was the vehicle for offerings to the deities. Ayurvedic texts began codifying medicinal uses of volatile plants: turmeric, neem, vetiver, sandalwood, for healing through inhalation, massage, and fumigation. In China of the Western Zhou and spring and autumn periods, till 1046–481 BC, bronze ritual vessels held wine infused with aromatic herbs. The fermented millet or rice wine carried yeasty, fruity notes amplified by added cassia, ginger, and peppercorns. Ancestor temples burned bundles of artemisia and milfoil during divination. The sharp, camphor green smoke helped induce the trance state needed to communicate with the dead. Across these civilizations, scent became economic, taxed, monopolized, forged, counterfeited, social, used to mark rank, royal versus commoner perfumes, gender, ritual, purity, medical, inhaled for respiratory relief, applied for skin conditions, used in embalming, religious. The language of sacrifice, purification, divine appeasement, artistic, incorporated into poetry, myth, theater, incense in Greek drama, scented oils in banquets, technological. Distillation experiments in Egypt and Mesopotamia produced early essential oils. Good smells evoked divine favor, royal power, health, fertility, memory of the dead, sexual allure, seasonal renewal, wealth. Bad smells, while still warnings of disease, spoilage, impurity, were countered with increasingly sophisticated masking, fumigation, and ritual cleansing. By around 500 BCE, the stage was set for the classical worlds of Greece, Persia, India, and China to inherit and further elaborate this scented legacy. Philosophers debating the nature of odor, physicians classifying smells by humoral properties, perfumers creating signature blends, traders risking lives across deserts and seas for frankincense and cinnamon. The nose had become not just a sense, but a cornerstone of civilization itself. The next chapter will follow that classical flowering and the dramatic transformations brought by Hellenistic empires, Roman expansion, Silk Road intensification, and the rise of New World religions that would forever reshape the cultural meaning of scent. We now enter one of the most richly documented and philosophically self-aware periods in the history of scent, the Classical Age and its long aftermath, stretching from roughly 500 BCE to about 500 CE. This 10th chapter follows the olfactory cultures of Greece and the Hellenistic world, the Roman Empire at its height, the Parthian and early Sasanian realms of Persia, the flowering of South Asian Ayurvedic and Buddhist traditions, the consolidation of Chinese imperial perfumery under the Han and subsequent dynasties, and the dramatic transformations brought by the rise and early spread of Christianity, late antique Judaism, Manichaeism, and the pre-Islamic Arabian incense trade. Across these 5 centuries, smell was dissected by philosophers, classified by physicians, commodified on an unprecedented scale by merchants, sacralized by New World religions, eroticized in literature and daily life, militarized in siege warfare and hunting, and turned into an object of luxury, nostalgia, and moral debate. The nose became both a scientific instrument and a moral compass. Odor was no longer merely lived. It was theorized, regulated, mythologized, and traded as never before. In the Greek world of the late Archaic and Classical periods, tilde 500–323 BCE, philosophers began to ask fundamental questions about what smell actually is. Empedocles and his followers argued that odors are effluences, tiny particles streaming off objects that enter the nostrils and interact with sensory pores. Democritus, the atomist, proposed that smell arises from differently shaped atoms that fit or clash with receptor channels in the nose, an idea remarkably close to modern lock and key receptor theory. Plato in the Timaeus described odors as vapors arising from wet substances, pleasant when they restore balance to the body, painful when they disrupt it. Aristotle devoted an entire treatise on sense and the sensible to olfaction, classifying smells into 2 broad categories, those that are intrinsically pleasant or unpleasant, and those that become pleasant or unpleasant only in relation to appetite. For example, the smell of food is delightful when hungry but nauseating when full. He noted that humans have a relatively weak sense of smell compared with other animals, yet possess the unique ability to appreciate odors aesthetically, divorced from immediate survival needs, a capacity he linked to our rationality. Physicians of the Hippocratic and later Hellenistic schools built practical taxonomies. Smells were humoral, sweet and aromatic odors, rose, saffron, myrrh were warming and drying, good for cold-wet imbalances. Sharp, pungent odors, onion, garlic, asafoetida were hot and drying, useful against phlegm. Foul, putrid smells indicated corruption and disease. Fumigation became standard therapy, burning resins, herbs, and animal substances to cleanse sick rooms, purify women after childbirth, or drive out miasma. Theophrastus, Aristotle's successor, wrote the first dedicated inquiry into plants that included whole sections on aromatic species: frankincense, myrrh, cinnamon, cassia, nard, costus, iris root, detailing their habitats, harvest, preparation, and characteristic volatiles. He distinguished true cinnamon bark from cheaper substitutes by smell and taste, already aware of adulteration in the luxury trade. The Hellenistic period after Alexander's conquests, 323–31 BCE, fused Greek theory with Egyptian, Persian, and Indian practice. Alexandria became the perfume capital of the Mediterranean. Perfumers Myropsoi compounded complex unguents in workshops near the Miocene. Mendesian ointment blended myrrh, cinnamon, cardamom, nard, honey, wine, and raisins into a thick paste. Metopian carried rose, crocus, cinnamon, and spikenard. Susanum featured lilies, myrrh, cinnamon, and saffron. These were sold in alabaster or glass vessels, often stamped with the maker's name. Women and men anointed themselves daily. Symposia were perfumed with incense burners and scented oils poured over guests' heads and feet. Theophrastus complained that excessive perfume use masked body odor rather than complemented natural scent, beginning a long moral debate about authenticity versus artifice. Rome inherited and massively amplified this tradition. From the late Republic through the Empire, 200 BCE 500 CE, the city of Rome itself was an olfactory palimpsest. Subura streets reeked of garum factories fermenting fish into pungent sauce, tanneries processing hides in urine vats, fullers cleaning wool with ammonia-rich stale urine collected in public latrines, bakeries exhaling yeasty sweet bread volatiles, street vendors roasting chickpeas and nuts over braziers. Patrician houses countered with atrium gardens planted with violets, roses, lilies, and saffron crocus whose dawn perfume drifted through colonnades. Elite women carried perfume flasks, aribal, and scent boxes. Men used solid unguents and liquid oils. Martial satirized the over-perfumed dandy whose scent preceded him like a herald. Roman perfumery operated on industrial scale. Imports arrived via the Red Sea Nile Alexandria route and the caravan paths through Petra. Frankincense and myrrh from South Arabia, cinnamon and cassia from South India and Sri Lanka via Muziris and Arakamdu, nard and costus from the Himalayas, Malabathrum cinnamon leaf oil from the Gangetic Plain, saffron from Corycus in Cilicia or Persia, iris root from Illyria, storax and galbanum from Syria. Pliny the Elder devoted Book 12 of Natural History to aromatics, lamenting that Rome spent 100 million sesterces annually on Eastern luxuries, half of it on perfumes. He listed dozens of compounds. Unguentum regalium blended dozens of ingredients for emperors. The famous Unguentum telinum mixed fenugreek, marjoram, iris, honey, and wine. Public life was scented. Theaters and amphitheaters were sprinkled with saffron water to mask the smell of crowds and animal blood. Triumphal processions burned incense along the route. Temples maintained perpetual braziers of frankincense and wine-soaked resins. Funerals used large quantities of aromatics, myrrh, cassia, nard, to mask decomposition during lavish processions. The pyre of Augustus reportedly consumed vast quantities of Arabian incense. Gladiatorial games saw spectators anointed with rose oil to counteract the stench of blood and sweat. In medicine, Galen and his followers expanded smell's therapeutic role. Aromatics were prescribed for headache, rose oil on the temples, fainting, burning feathers, or a saphitida under the nose, uterine disorders, fumigation with myrrh and bay, melancholy, saffron, and ambergris. Galen noted that pleasant odors lift the spirits by acting on the animal spirits in the brain. Foul odors can induce convulsions or syncope. In South Asia, the period saw the maturation of Ayurvedic perfumery and Buddhist ritual use of scent. The Karaka and Sushruta Samhitas, BC 200 CE, classify odors by rasa, taste like qualities: Madhura sweet, Amla sour, Kadu pungent, Tikta bitter, Cassia astringent, Lavana salty. Fragrant substances: sandalwood, agarwood, camphor, vetiver, jasmine, champaka, lotus, are cooling, balancing pitta. Pungent aromatics, asafoetida, hing, black pepper, stimulate digestion and dispel kapha. Attar's non-alcoholic perfumes were distilled in copper stills using sandalwood base oil. The famous Idar Ijat jasmine attar and Gulab rose water emerged during this era. Buddhist monasteries burned sandalwood and aloeswood in stupas and during pujas. The heavy, creamy, sweet smoke of burning sandal was believed to carry merit to the deities and calm the mind during meditation. Monks carried personal incense pouches. Pilgrims offered aromatic ghee lamps at Bodh Gaya and Sarnath. In China, the Han Dynasty, BC 220 CE, and subsequent Three Kingdoms and Jin periods refined imperial perfumery. Palace women used lead white face powder scented with orchids and musk. Emperors burned aloeswood, Chen Xiang, and dragons brain camphor in audience halls. The Book of Han records tribute from Southeast Asia, including liquid amber, storax-like resin, and golden perfume, ambergris or musk. Aristocratic women wore sachets of dried flowers, spices, and musk deer glands sewn into silk undergarments. Funerals used large quantities of aromatic woods for pyres. Elite tombs included bronze censers shaped as mountains with holes for rising smoke, symbolizing paradise islands. The Parthian 247 BCE 224 CE and early Sasanian 224 651 CE empires controlled the western end of the incense route. Ctesiphon and Seleucia on the Tigris were perfumed entrepots. Zoroastrian fire temples fed fragrant woods to sacred fires. The smoke purified the air and carried prayers. Royal hunts used musk and civet gland secretions to lure game or mark territory. As the Roman Empire Christianized, 312-500 CE, scent acquired new theological weight. Early Christians associated frankincense with the Magi's gift to the infant Jesus, myrrh with his burial. Churches adopted incense from Jewish temple practice. Swinging censers filled with frankincense, myrrh, and storax created clouds that symbolized prayers rising to heaven. Baptismal chrism was blended with balsam and olive oil. The sweet, resinous scent marked the newly baptized as Christ's anointed ones. Martyrs' relics were venerated with aromatic oils. The smell of sanctity, roses or violets issuing from graves, was taken as proof of divine favor. In late antique Arabia, pre-Islamic tribes burned frankincense and aloeswood in sanctuaries. The Kaaba in Mecca was perfumed annually with a special blend. The Quran would later praise good words as rising like perfume to God. Across the classical and late antique worlds, smell functioned as philosophical problem. What is odor ontologically? Medical tool diagnosis, fetor hepaticus, gangrene stench. Therapy, aromatherapy, avant la lettre, economic engine, driving desert caravans, Red Sea fleets, Indian Ocean trade, social marker, distinguishing citizen from slave, elite from commoner, pure from impure, religious language. The medium through which divinity was sensed and prayers ascended, erotic instrument. Perfumed lovers in poetry, Sappho, Catullus, Ovid, Kalidasa, military aid, smoke screens in sieges, scented arrows. Camphor for endurance, good smells evoked divinity, health, love, status, memory of the blessed dead, cosmic harmony. Bad smells signaled disease, moral corruption, demonic presence, social degradation, impending death. By around 500 CE, the classical olfactory order had crystallized, a global network of scent knowledge, trade, ritual, and theory that would be inherited, adapted, and dramatically expanded by the medieval worlds that followed: Byzantium, the early Islamic caliphates, Tang China, Gupta and post-Gupta India, and the emerging kingdoms of Europe. The next chapter will trace how the rise of Islam, the Silk Road zenith, the medieval spice obsession, and the alchemical distillation revolution turned perfume into high science, sacred art, and one of the most valuable commodities on earth. We now cross into the medieval millennium and the early modern threshold, roughly 500 CE to about 1500 CE, 1,000 years when the global story of scent became dramatically more interconnected, scientifically ambitious, religiously fervent, commercially ferocious, and technically innovative. This 11th chapter traces the olfactory civilizations of Byzantium and early medieval Europe, the explosive rise and golden age of Islamic perfumery and pharmacology from Baghdad to Cordoba to Delhi, the Tang Song Yuan refinement of Chinese aromatic culture, the maturing of Indian adder traditions under Gupta successors and Islamic sultanates, the Crusades as scent highways, the Mongol Silk Road zenith, the Black Death's olfactory trauma, and the late medieval European rediscovery of distillation that would soon birth modern perfumery. Across these centuries, smell was elevated to high art, sacred science, global currency, medical panacea, courtly language, plague marker, and spiritual currency. The nose guided pilgrims to paradise, physicians to diagnosis, alchemists to the quintessence, merchants to unimaginable wealth, and mourners through the stench of pandemic death. In the Byzantine Empire, 1453 CE, Constantinople remained the Western world's perfume metropolis long after Rome's fall. The imperial court maintained enormous stocks of aloeswood oud, ambergris, musk, rose water, and frankincense. Basil II's palace ceremonies filled the great hall with clouds of burning storax, benzoin, and sandalwood. The heavy, balsamic sweet smoke was said to make the emperor appear as an icon surrounded by divine fragrance. Byzantine perfumers perfected the art of solid pastilles, mixtures of resins, gums, powdered spices, and animal musks pressed into cakes that slowly released scent when warmed against the body or placed in clothing. Churches swung massive silver censers filled with kyphi-like blends during the divine liturgy. The incense was understood to make invisible the boundary between earth and heaven. When the Fourth Crusade sacked Constantinople in 1204, crusaders carried off tons of aromatics, myrrh, spikenard, camphor, rose attar back to Western Europe, seeding a medieval perfume revival. Early medieval Western Europe, 500–1000 CE, by contrast, lived in a sparser olfactory landscape. Monasteries preserved Roman and Byzantine knowledge in herbals. Monks grew rose, lavender, sage, hyssop, and rue in cloister gardens, using them for liturgical incense, medicinal fumigations, and to sweeten the air of scriptoria. The stench of unwashed wool, peat fires, tannery vats, and open latrines dominated daily life in towns, yet the church insisted on aromatic purity, chrism oil blended with balsam, altar incense of frankincense and myrrh when affordable, myrrh-soaked burial linens for saints. Pilgrims to Rome, Compostela, and Jerusalem returned with vials of holy earth or Jordan water, sometimes infused with local aromatics. The rise of Islam after 632 CE transformed scent into one of the most systematized, celebrated, and globally traded aspects of civilization. The Prophet Muhammad is reported to have loved perfume, declaring it one of the three things the world should enjoy, along with women and prayer. Hadith encouraged the use of musk, ambergris, aloeswood, and rose water. Early caliphs in Damascus and then Baghdad established royal perfumeries, Dar al-Idr. Under the Abbasids, 750–1258 CE, Baghdad became the world capital of scent science. Al-Kindi's book on the chemistry of perfume and distillations, 9th century, is the earliest surviving systematic treatise on the subject. It describes more than 100 recipes for compound perfumes, including the famous Baghdadi musk and Gallia, a blend of musk, ambergris, civet, aloeswood, saffron, rose, and camphor. Al-Razi, Ibn Sina, Avicenna, and later al-Biruni classified odors according to humoral theory, adding Islamic refinements. Sweet fragrances strengthen the heart and brain, pungent ones dispel phlegm, fetid ones indicate corruption. Islamic perfumery perfected hydrodistillation. Copper stills, alembics with long swan neck condensers produced true essential oils and floral waters on an industrial scale. Rose water from Nishapur, Shiraz, and later Kashan, orange blossom water from Seville and Damascus, jasmine attar from Kanauj and Fars, agarwood oil from Assam and Cambodia all became standardized luxury exports. Markets in Baghdad, Cairo, Cordoba, and Samarkand displayed hundreds of glass phials and ceramic jars. Perfumers compounded bespoke scents for caliphs, viziers, and wealthy merchants. Women carried musk or ambergris pastilles in filigree gold pomanders hung from belts or necklaces. These released scent gradually, creating a moving personal aura. Mosques and madrasas burned aloeswood chips on charcoal during Friday prayers. The creamy, woody smoke was believed to please God and calm the congregation. In Al-Andalus, Islamic Spain, 711–1492 CE, Cordoba and Granada developed a distinctive olfactory aesthetic. Gardens of the Alhambra and Generalife were planted with myrtle, jasmine, rose, citrus, and basil. Their combined volatiles created layered fragrance zones that changed with the hour and season. Poets like Ibn Zaydun and Walada wrote of lovers meeting under jasmine arbors where the scent became inseparable from erotic memory. When the Christian Reconquista advanced, captured cities were ritually fumigated with frankincense to purify them of Muslim presence, yet many Christian nobles quietly continued using Andalusi adders. In India, the post-Gupta and early medieval period, 500–1200 CE, saw the efflorescence of adder making in Kannauj, Uttar Pradesh, which remains the subcontinent's perfume capital. Sandalwood-based distillation produced concentrated itters of champaka, cura pandan, vetiver, saffron, and henna. Buddhist and Hindu temples burned massive quantities of sandalwood and camphor in artis. The thick, milky, sweet smoke was thought to carry offerings directly to the deities. Islamic sultanates after 1200 CE blended Persian, Arabic, and Indian traditions. The Delhi Sultanate and later Mughal courts adopted Gallia and invented new blends featuring patchouli, agarwood, and rose. Tang China 618–907 CE and Song China 960–1279 CE brought imperial perfumery to new heights of refinement. Palace women wore sachet belts filled with dried orchids, cloves, aloeswood shavings, and musk. The scent rose gently as they moved. Emperors burned dragon's saliva, ambergris, and Chen Xiang aloeswood in audience halls carved to resemble mountain landscapes, the smoke drifting through holes to mimic mist. Song scholars compiled pharmacopoeias listing hundreds of aromatics. Li Shizhen's later Benkao Gangmu would systematize them. The Mongol conquests 1206–1368 CE linked China directly to the Islamic world. Marco Polo described the odoriferous woods shipped from India and Southeast Asia to Symbolic, Beijing. The Crusades 1095–1291 CE acted as massive scent exchanges. Frankish knights encountered Eastern perfumes in Antioch, Tripoli, and Jerusalem. Many returned with rose water, musk pomanders, and recipes. The Knights Hospitaller and Templar maintained pharmacies that compounded aromatic medicines. The Mongol peace, Pax Mongolica, 1250–1350 CE, turned the Silk Road into the greatest olfactory highway in history. Caravans carried frankincense from Dhofar, ambergris from the Somali coast, musk from Tibet, aloeswood from Laos, cloves from the Moluccas, nutmeg from Banda, all converging on Tabriz, Samarkand, Bukhara, and Kashgar. The Black Death, 1347–1351 CE, created one of history's most traumatic olfactory chapters. Cities reeked of gangrene, vomit, diarrhea, and mass pyres of quicklime and sulfur. Physicians wore beak masks stuffed with cloves, cinnamon, lavender, rose petals, camphor, and ambergris to filter miasma. Streets were fumigated with juniper, rosemary, and vinegar-soaked rags burned in braziers. The smell of plague became a cultural trauma. Survivors associated certain pungent herbs with survival and safety. By the late Middle Ages, 1300–1500 CE, Europe rediscovered distillation through Arabic translations. Monks in Salerno and later Venice produced aqua mirabilis, marvelous water from alcohol and aromatics, early eau de cologne precursors. Guilds of apothecaries and perfumers emerged in Paris, Venice, and Florence. They compounded Hungary water, rosemary spirits, and eau de la Réunie for royalty. Ambergris washed ashore on Atlantic beaches was worth more than gold. Musk pods from Siberian and Tibetan deer were smuggled across Asia. Across this millennium, smell functioned as Sacred medium, mosque, church, temple smoke carrying prayer, medical cornerstone, distilled oils for plague, melancholy, childbirth, courtly language, personal scent as signature of rank and taste, global currency, frankincense, musk, ambergris traded like bullion, plague marker, feeder of disease versus protective, herbal clouds, alchemical goal, the quintessence extracted by distillation, erotic and poetic motif. From Andalusi guzzles to Persian masnavis, good smells evoked paradise, Islamic Jannah gardens of musk and saffron, divine presence, health restored, royal favor, erotic intimacy, spiritual elevation. Bad smells, now inescapably linked to pandemic death, were countered with ever more sophisticated masking, fumigation, and ritual purification. By around 1500 CE, the stage was set for the early modern explosion. Portuguese rounding Africa to tap Indian Ocean spice routes directly, Ottoman control of the Levant intensifying European demand for Eastern aromatics, the scientific revolution questioning Galenic smell theories, and the birth of true alcohol-based perfumery in grass and cologne. The next chapter will follow how the age of exploration, the scientific revolution, the Enlightenment, and the Industrial Revolution turned scent into a mass-produced luxury, a chemical industry, and a subject of modern psychology, while colonial plantations reshaped the global geography of fragrance forever. We now reach the final arc of this long journey, from roughly 1500 CE to the present day. This 12th and concluding chapter sweeps through the age of exploration that shattered old scent monopolies, the scientific revolution that turned smell into chemistry, the Enlightenment and Romantic eras that made fragrance a language of emotion and individuality, the Industrial Revolution that democratized perfume for millions, the birth of modern synthetic perfumery, the rise of global fragrance houses. The psychological and neuroscientific understanding of olfaction in the 20th and 21st centuries, and the contemporary world in which scent is simultaneously a multi-billion-dollar industry, a tool of memory science, a marker of personal identity, a weapon in marketing, a therapeutic modality, and for many, a quiet spiritual practice in an increasingly deodorized, sanitized, yet strangely scent-hungry age. When Vasco da Gama rounded the Cape of Good Hope in 1498 and Portuguese carracks began docking directly at Calicut, Cochin, and Malacca, the centuries-old Islamic and Venetian middlemen were bypassed. Cloves, nutmeg, mace, cinnamon, and pepper flooded Lisbon at prices that eventually fell by factors of 10. But the real olfactory revolution came with the Portuguese and later Dutch and English capture of the Moluccas, Spice Islands, and the Banda Archipelago. Nutmeg and mace trees were monopolized, guarded with forts and massacres. The scent of freshly grated nutmeg, warm, sweet, woody, almost narcotic, became a literal smell of colonial profit. In Europe, the sudden abundance of these spices saturated food, medicine, and perfumery. Clove water and nutmeg pomanders appeared in apothecary shops. Ambergris, still gathered from Atlantic beaches, was now compounded with New World imports, vanilla from Mexico, tonka beans from Venezuela, cacao, whose roasted bitterness began appearing in elite chocolate-scented perfumes. The 17th century saw the true birth of modern alcohol-based perfumery. In 1709, Johann Maria Farina created the first recognizable eau de cologne in the German city of Köln. An alcohol tincture of bergamot, lemon, orange, neroli, lavender, rosemary, and petitgrain whose fresh, citrus herbal brightness contrasted sharply with the heavy animalic, resinous oriental blends of the past. Farina's formula was immediately copied across Europe. By the 18th century, cologne became a generic term for light, refreshing eau de toilette. Grasse in Provence emerged as Europe's flower-growing capital. Fields of jasmine, rose centifolia, tuberose, violet, and orange blossom were harvested at dawn when volatile release peaks, distilled into concretes and absolutes that supplied the growing houses of Paris and London. The Enlightenment brought scientific scrutiny. Chemists began isolating active principles. Carl Wilhelm Scheele extracted citric acid from lemons, 1770s. Nicolas Leblanc produced synthetic soda for soap making, 1791, which indirectly supported the hygiene revolution that made strong body odor socially unacceptable among the middle and upper classes. The Romantic era romanticized scent as an intimate, emotional language. Goethe wrote of the invisible yet powerful way odors evoke memory. Baudelaire's correspondences equated smell with synesthetic transport to other realms. Perfume became a private poetry worn on skin. The 19th century Industrial Revolution turned fragrance into mass product. François Coty, Ernest Beau, Jacques Guerlain, and others built empires. Coty's L'Oregon 1905 and Chyper 1917 introduced novel accords, mossy, woody, aldehydic, that could not have existed before synthetic chemistry. In 1882, coumarin from tonka beans was synthesized. In 1888, vanillin was produced cheaply from guaiacol. By the 1890s, ionons recreated violet and orris notes at scale. Grass still supplied naturals, but synthetics allowed consistency, affordability, and entirely new scent families, Fauger, aldehydic, oriental, Chyper, that defined 20th century perfumery. The 20th century saw psychology and neuroscience finally explain what the nose does. Sigmund Freud linked olfaction to repressed sexuality. Later researchers discovered the direct limbic pathway. Odor molecules bind to receptors in the olfactory epithelium. Axons project straight to the piriform cortex and amygdala without thalamic relay, explaining why smells trigger instant, vivid memories and emotions. Linda Buck and Richard Axel's 2004 Nobel-winning work identified ~400 functional human odorant receptor genes and showed how combinatorial coding allows us to discriminate trillions of smells. Brain imaging revealed that familiar childhood scents, freshly mown grass, Crayola crayons, Play-Doh, activate the same hippocampal regions as Proust's Madeleine. World wars accelerated synthetic innovation. Shortages of naturals led to replacement molecules, but also trauma. Trench soldiers associated crassilic disinfectant with gangrene and death. Post-war society embraced clean, soapy, powdery fragrances as symbols of renewal. Chanel No. 5, 1921, with its massive aldehydic top note, became the first perfume marketed as a modern woman's signature scent. The mid-century saw the rise of masculine Faugers, Brut, Aramis, Aquatic's Cool Water 1988, and Gourmand's Vanilla Dominant Angel 1992. By the 1990s, celebrity fragrances, flankers, and limited editions turned perfume into a fast fashion accessory. Today, we live in the most olfactorily complex era ever. Global fragrance is a $50 billion industry. Synthetic molecules, iso-E super, ambroxan, cashmere, helvetalide, create transparent woody, amber, musky textures impossible with naturals alone. Niche houses, Serge Lutens, Frédéric Malle, Amouage Byredo, Le Labo revive rare naturals, oud from sustainable plantations, ethically sourced ambergris, iris butter from Tuscany, while biotech companies ferment vanillin and patchouli in yeast vats. Scent marketing saturates retail. Hotels pump signature fragrances into lobbies, casinos use calming lavender blends, supermarkets deploy fresh bread volatiles at entrances. Aromatherapy has gone mainstream. Essential oils treat anxiety, insomnia, nausea. Neuromarketing firms test consumer response to ambient scents in stores. Alzheimer's patients respond to personal scent cues, grandmother's perfume, pipe tobacco, when words fail. Yet we also live in an age of olfactory deprivation. Air conditioning, HEPA filters, antibacterial soaps, and fragrance-free policies strip everyday environments of natural odor complexity. Many urban children grow up never smelling a forest floor after rain, a barn after milking, or a kitchen after baking bread. At the same time, nostalgia-driven revivals, artisanal incense, natural perfumery, scent libraries, attempt to recover lost olfactory heritage. Indigenous knowledge systems, Amazonian shamans using tobacco and ayahuasca smoke, Aboriginal Australians burning eucalyptus for healing, remind us that scent remains spiritual technology in many cultures. From single-celled chemotaxis 3.5 billion years ago to the trillion scent discrimination capacity of a modern human nose, the journey of olfaction has been one of ever-increasing mastery. Good smells have always drawn us toward nourishment, safety, love, memory, the divine. Bad smells have always warned of poison, decay, danger, moral corruption. What has changed is scale, intention, and meaning. We no longer merely sense odors. We design them, bottle them, weaponize them, therapize them, commodify them, and use them to tell stories about who we are. And so the story closes where it began, in the nose, that quiet, ancient organ still whispering truths the eyes and ears cannot reach. Thank you for joining us on this long journey through time, from the chemical cues of ancient oceans to the perfume counters of today. If this documentary has stirred your own memories or made you pause to really smell the world around you, we'd love to hear from you. Please subscribe for more deep explorations of human history and the senses. Leave a comment below telling us your favorite scent memory or the smell that instantly takes you back to childhood. And let us know in the comments where in the world you are watching from right now. We read every one. Until the next story, keep breathing deeply.